2, that is Biosafety Laboratory 2 facility, okay, which are there in certain universities and also in research institutes and all the diagnostic laboratories. So COVID-19 though is a risk group 3 pathogens, but its diagnosis comes under BSL-2. It is not that risky. So or any laboratory, any molecular laboratory can do diagnosis for COVID-19. Risk group 3 pathogens that cause serious diseases but do not ordinarily spread from one individual to another. So the risk to individual is high, but not to the community. It won't spread or it will not cause an epidemic. Like KFD. Now, if you all are from Karnataka, if you all are Kannadigas, you must have heard of this disease or at least know this place called Kstacademy. It's a forest in Shivamogga. And this virus was identified in this forest. It is a zoonotic virus. It spreads from monkeys to humans. And this outbreak still happened in Kaisanur. It's a forest on the western Ghats of Karnataka. And even now these outbreaks do happen where humans get infected, and the mortality rate is around 5 to 10% case fatality rate. So this disease causes it causes hemorrhagic fever, and in the second stage, in the later stages, it may cause neurological disturbance like encephalitis. Okay, so KFD is it is caused by ticks. Ticks, you all are, those who have pets, they would know that ticks are parasitic insects. They feed on the blood of mammals like dogs, animals, and even humans. So these ticks from coffee plantations or people working in the farms and all, when they get tick bites, they get this, and if the tick is infected with this virus, they will get KFD. So it requires ticks for the transmission. So if a person is infected, if I am infected, I will not spread it to another person by close contact or by contact with fluids or anything. It is transmitted only through ticks. So that's why the risk to community is less, and hence it's classified as risk group 3. Why risk group 3? Because its case fatality is high and no preventive measures are available for KFD. Risk group 4, this is the most dreaded group of pathogens. They are pathogens that can cause severe diseases and are easily transmissible, and no preventive measures are known for it. Okay, that one example is Nipah, and you all know how scary is Nipah. Why? Because the case where one person gets infected, you must know of the Kerala incidents in 2018. One person got infected, it led to infection of 19 others in just a span of 2 days, and the case fatality is almost 80 to 100%. You get infected with Nipah, there is 100% chances that it may be a fatal infection, right? So such cases are risk group 4. You have to handle them in a BSL-4, Biosafety Laboratory 4 contained. So what is BSL-4? It is a highly contained laboratory. It is contained, it is like guarded. You cannot, the pathogens won't leave outside this laboratory. Nothing from the outside can go inside this laboratory. So the facilities is highly secured and highly contained. The containment is when you restrict the movement of the pathogen. Okay, so such facilities also in India, the only BSL-4 laboratory right now is in National Institute of Virology, Pune. Okay. So, yeah, so that was risk group 4 pathogen, right, like Nipah. So based on this, so if we know what the pathogen is, we will know what the risk group is, then we can easily handle them in BSL-2, 1, 2, 3, and 4. Now, there are certain cases like for COVID-19, we didn't even know what the pathogen is. We didn't know what is causing this. So what do we do? So any unknown pathogen, any new pathogen, any new epidemic will always be handled as a risk group 4 because we don't know what can it cause. So COVID-19 was first handled as a risk group 4 pathogen. It is still for research purpose handled as an RG4 pathogen, but for diagnostics and all, once we know, once we have the preventive measure, once we know its transmission route, how can it be transmitted, how can the transmission be prevented, now it is handled for diagnostic purpose in BSL-2 laboratory. So emerging and re-emerging viruses. Now these are the risk group pathogens. Now coming to what are emerging and re-emerging viruses. Now emerging and re-emerging infections are there for is here. What are emerging diseases? Newly identified and previously unknown infections when they cause an outbreak. What is an outbreak? Outbreak is when the infection is new, or the infection, if it's already there, the number of infected cases are more than usual, what usually happens. That is an outbreak. So emerging diseases are newly identified and previously unknown infectious agents. They cause public health problems either locally or internationally. So if it's national level, what do we call it? It's an epidemic. When it goes global, like COVID-19, it becomes a pandemic. That's a very red alert situation. Re-emerging diseases, infectious agents that have been known for some time, means they are in the circulation, it's in circulation in the environment, but they have fallen to such low levels, no longer considered as a public health problem, but suddenly they show a upward trend, and there is incidence of prevalence worldwide. And it also sometimes it happens that it was known in, so like KFD I just spoke about, it is known to be prevalent in Shivmoga or in districts around Shivmoga, basically in Karnataka. But suddenly in 2000, I think it was 20, there were KFD cases reported from Maharashtra. Then there were few KFD cases reported in Goa. Those are re-emerging cases. Okay, so where they are known to be endemic or known to be present in one particular locality, so the health authority over there is ready to handle it. But when it happens to a newer location, that is when it has to be taken seriously. So those are re-emerging diseases. So what is a global? These are the list of all the emerging and re-emerging diseases globally. You can see there are such infections are all around the globe, but mostly they are present in the tropical. Is it working? No. Okay. So, but you can see that it is mostly prevalent in the tropical areas of Asia and Africa. There are more cases over there, maybe because of the environment, the population, the cultural habits. Okay. So you see more emerging and re-emerging infection in these continents. Now this list has both all risk group pathogens, risk group 1, 2, 3, and 4. So in 2018, WHO, this was before COVID. So WHO, by then, because of the outbreaks that were happening of influenza, of Ebola, so that time, from that moment itself, the global health fraternity had noted down that we need to make some adjustment or some guidelines to deal with such upcoming infections or upcoming pandemics or epidemics. So WHO had classified, or they had launched a list of high risk pathogens. So why did they launch this? It was telling all the countries or telling all the regions that, see, these are the high risk pathogens that you have to look for. So you make your health system prepare to deal with this. How do you make them prepare? Your research and development, your diagnostic innovation should all focus on this. You should not wait for these pathogens to strike you. You should be prepared to deal with them as and when they arrive, right? So this was they made a list. So these are viruses from different families, like viruses, arena viruses. So many of them you must not have even heard of, but they are risk group pathogens, like the Lassa virus. If you can look at the clinical symptoms, all of them are fever, but there are different classifications of fever. The ones they call the severe respiratory symptoms and pneumonia, like SARS-CoV-2, So these fringe areas, these human rural, basically they are rural places. So their agricultural practices, they start they remove the forest area, they deforest the area and start using them as agricultural land. So you must have read in the newspapers in Kerala there was pig attack in the wild pig or wild boar attacks in the farm. Why? Because the farm was built in a forest area. So obviously there'll be elephants attacks, there is wild boar attacks, right? So they don't understand when this wild boar entrance or these wildlife enter into the human dwellings, even if they're not attacking the humans personally, but even if they're leaving the droppings or even if they're eating fruits or touching this something some of their body fluids are in the soil. So all these are sources of infection. These are examples or these are incidences which can cause spillover. What's a spillover? Spillover is when the pathogen gets transmitted from one species to another. So when the pathogen gets transmitted from the wildlife to human, from the domestic to human, okay, that's called a spillover. So these are all instances where spillover can happen. Then viral factors are also a cause. There is genetic evolution and recombination happening and vector-borne diseases. So even not just the reservoir, even if the vector habitat is disturbed, mosquitoes, the more and more you go into, forest is like a hub of mosquitoes. The more and more you go into the forest, the mosquitoes are coming to your urban sites. Like from the rural, it is also coming to your urban settings, right? So now studies have shown that mosquito species which were in the forest are now quite prevalent in the urban settings also. Like the Aedes mosquitoes, Aedes species which were found only in the forest, now you can also find them in the cities. So when they are coming to the cities, they are also carrying them with them all the infections that was there in the forest. Right? So the habitat of the vector is also one of the factors for zoonotic infections. And immunosuppression. If the population is not healthy, they'll be if they are more immunosuppressed population, then they will be more susceptible to the infection. Advancement in technology. So I guess all of them are from science field, so I don't need to explain what is whole genome, what is next generation sequencing, right? So everyone must have heard of it now, at least not in your theory, but the COVID-19, during the COVID-19 era, you must have heard of next generation sequencing, right? This is one technique which help us identify SARS-CoV-2 and also its variants every time it causes an epidemic, right? So all these new advances, because of all these new advanced technologies, we are able to detect these infections more efficiently than we were able to do it with the traditional methods, right? So people say that these infections or these viruses were present earlier also, but we were not able to study them or identify them because we didn't have the tools then. Right now, we have the tools, so they are getting identified. So some people say in West Bengal in 2001, the Nipah case was identified. Later on, how come it was not, later, I think it's only in 2018 that it struck in Kerala. So like why it took so long to cause an outbreak. So there were cases, there are school of thoughts which say that we didn't have the tools to detect Nipah viruses as efficiently as we have it now, right? So that's why these infections or the outbreaks are getting more notified to the public. So as I said, travel is one of the major causes, major factors causing causing the emergence and re-emergence of infection. So you must be very fascinated to know. Even there are projects going on where they study mosquitoes that travel in the train. In India itself, we have projects where they collect every time a train comes to the station, they have their study sites. Every time the train comes to the station, they'll go in the train, collect the mosquitoes from the train. Yeah, and they'll study what is the origin of those mosquitoes, where have they traveled from. Okay, so travel is of important. Then migratory birds. You all must have heard of avian influenza, right? H5N1. So that is one example. Even migratory birds have other infections, reservoirs for other infections. But avian influenza is the top priority of zoonotic infection because of its transmissibility and the symptoms it causes. So avian influenza has not caused a human infection yet, but it is under the radar because it's an RNA virus and it can RNA virus. So there are different classes of viruses based on the genome material. There will be RNA virus, single-stranded RNA, double-stranded RNA, positive sense RNA, minus strand RNA, and then there are also DNA viruses. Of all these classes, we always are worried about the negative sense RNA virus. I have a slide on KFT ahead where I've given the replication, the pathogenesis of the virus. There I'll try explain why we are more worried about RNA viruses. So avian influenza is an RNA virus. It has more potential for mutation, and influenza is known to mutate every time. So I don't know how many of you all are aware. Influenza virus, the vaccine for influenza, common cold. It's not avian influenza, it's influenza which is there in humans, which is an human infection. Avian influenza is a bird infection. Human influenza, that is you all get common cold, right? Someone just coughed. So that's human influenza. So, and there are vaccines available for human. It's not under the immunization program. It depends on the medical practitioner. They may advise you to get an influenza vaccine depending on the season, depending on your health status. He may or may not advise you to get an influenza vaccine. It's not a mandatory vaccine. So this vaccine, it is not, so this year's influenza vaccine is not the same the next year. It's not the same the next. So every year the vaccine comes from WHO. Every year this vaccine is different. What is different? The strains in these vaccines are different. Why? Because influenza is a rapidly mutating virus. And so the vaccine that is available this year is based on the strains that are circulating in your country or in your region in this year. So next year again, so who tells the WHO team that this is a virus? There are surveillances going on. So there are laboratories which are every time surveilling, monitoring which is the influenza infection in the currently in circulation, which is the strain of infection that is current, and they report to WHO. Based on that, they develop the vaccine. So every year this vaccine changes. So, it's not like the same vaccine. So this tells the importance of mutations in the viruses. So migratory birds are very important for carrying such RNA viruses like the avian influenza. So why do we need to be prepared for these epidemics or pandemics? We all seen what happened in COVID-19, right? COVID-19 is one case which happened on a large scale. That's why people are aware of it. But there have been outbreaks happening before that also, Ebola. But it was more contained or more not at a global level. Because COVID happened in a global level, it came to the masses. Even the common public became aware of it. So why do we have to be worried about this epidemic or pandemic? Can anyone tell me? Why the government has to be prepared for the next pandemic? Sorry? To prepare the vaccine, that's one way of being prepared for it, right? To stop the spreading of the virus, yeah, before it becomes a large scale infection, you be prepared so that you can contain it, or nip it at the bud, like you say in English, nip it at the bud. But why? Because every pandemic has its own problems. It causes political instability, economic issues, loss of employment. You know how many people lost their life. Loss of employment, crime. There are articles which say crime levels increased after COVID-19. Why? There's no employment. People have lost their job. There is no work. Then quality of life reduces every pandemic, right? Though it may not be fatal, people who get infected, there's something called as long COVID, right? So people who got infected with COVID-19 and recovered, it was not after 2 to 3 years also, they were showing symptoms of breathlessness or fatigue, tiredness, muscle weakness. These are all it's called sequelae. After the infection is recovered also, you show some symptoms. So there are many infections which cause sequelae, but COVID-19 caused major sequelae. So every pandemic, this pandemic or epidemic will reduce your quality of life. It causes a burden on your health services also. If you are not prepared for a large scale to handle a large scale pandemic, causes a burden on the health facility, causes a burden on the government, the economy of the burden, right? So to avoid all that, government realized that we have to be prepared before the outbreak for handling such situations. Okay. So how do we be prepared? One approach that is like the trending theory is One Health approach. Okay. So this One Health approach is currently trending, but it was professed right in I think 1800s. It was said by some German researcher that he noted that between animal health and human health there is a very fine line. He said, okay, he said it in right in 1800, but no one. I mean, people. It was like a normal veterinarian saying something. But what they meant, what it led to, or what was the theory behind it is like, if you have to tackle human infection or human health, if you have to safeguard human health, you have to safeguard the animal health, the environment health, because all these are interconnected, right? So I said this is zoonotic infection is one of the major proportion of infections that is bothering humans, right? So only if you take care of the animals, or if you monitor the animals, will they not come to you. So that is one level of preparedness. You take care of your animal health, take care of your environment health, then indirectly you're taking care of the human health. So that is one health for all the sectors. Health, the health word should be common, which each and every sector should be taken care of. So One Health is an integrated transdisciplinary approach which recognizes that health of human, animal, and environment is interlinked and interdependent. This approach aims to sustainably balance, optimize health of human, animal, and ecosystem, because sometimes it's not directly from the animals to humans, it is also through the ecosystem, through the environment. So in this figure you can see the three sectors, the environment, human, and animal sector, and over here you can see the ecosystem, the healthy ecosystem, healthy animals, healthy humans, how they are interconnected. And this approach, how will this approach be attained? By collaboration of all sectors. So environmental scientists have to collaborate with the veterinary scientists have to collaborate with the clinical scientists. So this multi-sectorial collaboration, communication, and capacity building. You train people of all the sectors to handle such pandemic kind of situation. So this is One Health approach, and such an approach should be applied not just in the urban or at the national level, even at the regional level, even at the rural level, even at the smallest unit of your country. One Health approach should be applied. That is, all the sectors should work together, or should be it should be a transdisciplinary, a multi-sectorial communication, right? So this is not something which is only in the theories. It is implemented right now. It's being implemented very severely or seriously. So what are the key action points in One Health concept? Surveillance of early detection of the infection in the environment, strengthening of laboratory infrastructure. So your country should have the laboratories to handle these infections. If it's a risk group 4 pathogens, your country should be equipped with the laboratories to handle it. This shouldn't happen. There is a pathogen, but you cannot detect it. You have to send the samples outside. No? That can lead to delay in the diagnosis, which can lead to delay in treatments. Trained workforce. Laboratory should be there, and it should be workforce which are able to handle these pathogens, or to use those laboratory infrastructures, right? Strengthen healthcare, stakeholder engagement. As I said, One Health. Different sectors or different stakeholders should come together to address the issue, right? It's not that it's a human infection, so only the medical health sector will look after it. No, because these human infections are zoonotic, even the animal sector stakeholders should participate in dealing with it, right? In making policies regarding these infections, right? Strong research infrastructure, appropriate governance, political support, and international collaborations. So now we have to deal. We are a middle income society, right? A developing society. So there are facilities which are yet to come to our country, though we have come a very far way from what we were a decade ago. But international collaborations are important to train us for our capacity building and also for infrastructure development in our own country. So when I say early detection, how early? When? What is this early detection? This is all in terms of zoonosis, because zoonotic infections are the emerging and the re-emerging infections. So when I say zoonotic infections have to be detected early, when? So you see in the figure up, before early detection, you have to compare the figure up and with the graph down, the graph up with the. So you see when if you're able to detect the pathogen when it is in the reservoir, okay, that is in the animals, it is called forecasting the disease. Means the disease has not come to humans, but you know it's there in the animals. It's called forecasting. So that is also done right now. How do you do forecasting? Do you know what is forecasting and how do you do forecasting of a disease? So this is something called as modeling, mathematical modeling. I think there is an AI. I don't know. I'm not sure whether it will be dealt with. So it's all deals with machine learning, artificial intelligence, and machine learning. So you know there's a disease, like if I take avian influenza, you know it's an emerging disease. It's not so it's a re-emerging disease, not a new infection. So you know everything about its pathogenesis. You know which are the factors involved in there is migratory birds, when they migrate, what kind of ecosystem the birds need to migrate, and what kind of population is more susceptible to avian influenza. Everything is known. How many in each outbreak, how many people get infected, that is known. What are the everything. So all these factors, if you put them together in a machine learning formula or mathematical formula, it will generate a predictive model. It will generate a predictive model. What is a predictive model? It's nothing but a formula, mathematical equation, such that you put in every season or every year, you put in these factors in a value, and it will tell the predictive model can predict whether you will face an outbreak or no. So that is called forecasting. So that happens when the pathogen is still in the reservoir stage. But if the spillover happens, if the pathogen crosses from the reservoir to the domestic life or domestic animals or directly to the humans, if you can detect the pathogen at that spillover stage, that is early detection, right? So right now we are the government is putting everything together for having this early detection in place. Okay, right at the spillover stage, we have to detect that there is an infection, a major infection that's coming in the future. But if you come to this red, if the detection happens in that red peak, that is when it's already in the human and it's amplified in the human, then that's too late. Then it will be a pandemic, COVID-19 like situation. Okay, so our aim is to have an early detection during the spillover stage, right? So this is one of the principles of One Health. How will we have this early detection? So that's called a One Health cell. What is surveillance? Monitoring. Okay, so it is not like there is an infection, and if there are some cases, some humans reporting some disease, some fever, and they go to the doctor, the medical practitioner. Medical practitioner suspects that something is wrong. He asks them to do some test, monitor. It comes positive. Then he reports to the authorities. Then the authorities report to the higher authority, and then they realize, oh, there is an infection. No. Surveillance is continuous monitoring. You keep monitoring every time the public health authority is reporting, the medical practitioner comes to know that there's a positive case, it directly goes to the national level. That's surveillance, and continuously monitoring it. This was symptomatic clinical surveillance, but now it is One Health surveillance. You just don't do monitoring of infected individuals or individuals showing some symptoms. You do surveillance of the animals. You do surveillance of the environment. You see what are these animals infected with. Okay, what are the pathogens, the high risk pathogens present in the environment, right? So that is One Health surveillance. That is so this is an example. This is slaughterhouse. You know what is a slaughterhouse, right? Right, where the meat producing animals are butchered. So there are government slaughterhouses. What you all see at the roadsides are small level slaughter. It's not slaughterhouses, there are butcher houses. So where small level private ones where they'll have few animals butchered and all. But the government has set up government slaughterhouses where around 600,000 every day on a daily basis, thousands of animals are butchered, and from there the meat gets distributed to other centers. So those are government slaughterhouse. So these are the regions which are where there is high chances of human animal and environment interface happening, interaction happening, right? So the ICMR specifically have identified such centers where which has the most highest potential of human, animal, and environment interface. Slaughterhouse is one. Then can you all name any other place like slaughterhouse where humans are in close contact with the animals? Zoos, very good. Any other? National reserve, national parks, right? Right, birds, what she says, right? Bird sanctuaries, national parks, zoos, all these have become one project, one multinational, national multi-sectorial project. So the slaughterhouse is one project. So where there are different, it's a One Health project. Means the slaughterhouse have been mapped in different regions of the country, and for each center, each mapped slaughterhouse, there is veterinary team, there is a clinical team, and there's an environmental team working on monitoring the infections in that particular slaughterhouse. So similarly, the projects are there for zoos. So what do? They will, on a regular basis, on a periodic basis, they will collect samples. Now, human samples, right now the human samples are restricted to only symptomatic cases. Only if the slaughterhouse workers report some disease or some illness, then the samples will be collected. So those samples, animal samples, before butchering only, periodically, some animal samples are collected. Similarly, environmental like water samples, the wall swabs, the floor swabs, where they are cutting, butchering the animals, where they're cleaning the animals. Okay, the drinking water of the slaughterhouse. So such projects are ongoing, and we have identified zoonotic infections which are prevalent. And it's not just important to identify zoonotic. Okay, there is a slaughterhouse worker which has reported some infection, some symptoms, and we do a diagnosis of that case, and we detect it is now generally there are bacterial, viral infections are also there, but if we say we have detected zoonotic infection, suppose I'll go with bacterial because I remember there are more bacterial infections that were detected. Brucellosis. Brucellosis is a very prevalent zoonotic infection. It is very common in the slaughterhouse. We detected brucellosis, brucella. Okay, it's a bacteria. So, okay, it is present. Then we detected that the brucella is also was detected in the animal samples, right? The animal samples. And so, okay, fine, they're present. Now what? No, that doesn't say that there was a spillover. That doesn't say that the human that was infected was from that animal that we detected, right? How do we match that? For that, we do NGS. First, we do a time and space analysis. We see when was the animal infected, when was the animal came positive, and brucella has an incubation period of 1 to 2 months. So whether that human was also infected within that 1 to 2 months. If not, then we cannot say that's an event of spillover, right? Second confirmation is we do an NGS. We say whether the brucella strain that was identified from the animal is the same strain that was identified from the humans. Okay? So these kind of One Health studies are being carried out. And NGS, that is whole genome sequencing, plays a very crucial role in these studies. So as I say, pandemic preparedness, infrastructure development was one goal. So this was about surveillance, early detection, then is infrastructure development. So I'm going to tell about the infrastructure development done by ICMR because I work for ICMR. So we, after COVID-19, this happened specifically after COVID-19, there were around 139 virus research laboratories that were developed all across India. That map shows the location of all the 139 BRDLs. 16 BSL-3 laboratories, BSL-3 biosafety level 3 laboratories which handle biosafety level 3, risk group 3 pathogens, risk group 3 and to some level risk group 4 pathogens. Then 52 next generation sequencing sites, 3,300 molecular testing labs, indigenous molecular mobile BSL-3 laboratory. We have one mobile indigenous. It was prepared. It was developed and set up by in India. So it's an indigenous mobile BSL-3 bus. So it is a laboratory on the move on wheels. So there's many of these outbreaks happen, as I said, in the rural setup. So by the time the samples are collected, the contacts are traced, and their samples are collected and then sent to NIV Pune, there will be some time that is lost, right? So what we do is, if the moment there is some suspicious cases out, the mobile BSL-3 is deployed over there. So that so the samples from the suspected cases, animal samples, contact cases, everything gets tested in this mobile BSL. So it's fully equipped like a BSL-3 laboratory. So you can see in that small panel, the inside of the bus is also seen. Then there is one health center that is being established at Nagpur, and the government realized during COVID-19 how much important virology laboratories are. So apart from NIV Pune, there are 4 new NIV-like institutes that is being proposed right now. One in the north in Jammu, south Bangalore is going to get a South Zone National Institute of where I come from is a field unit. So we're going to get a South Zone National Institute of Virology Institute, okay, in Hebbal. Then there is one in Dibrugarh, that is northeast, and one in the central zone of India, that is Jabalpur. So I'll just go through it because we're almost out of time. So as I said, KFT is a zoonotic infection, very prevalent in Karnataka. It is spread through ticks. But how does it spread? Which animal reservoir? Monkeys. They are the main host of KFT. And it causes fatal infections in monkey also. So the moment in Casador forest, every year, it's a seasonal infection. Every year there are cases in Shivmoga and the adjoining district. Every time they spot a dead monkey in the fringe area or in the forest, the authorities are alerted. They will come, they will take the samples of the monkeys, they will test it in their laboratory. If found KFT positive, again the whole awareness and the whole surveillance starts. Okay. So it's a risk group 3 pathogen where it's handled in BSL-3 and 4 laboratories. As a positive sense, single-stranded RNA virus. I won't go in the detail of the genome and all. So this is a cycle of KFT. You can see there is tick which feeds on monkeys, and if it enters the human dwelling, it will also feed on the cattle and the domestic animals. Okay. The life cycle of the tick is shown. The unfed larva, the larval stage of the tick is generally present it doesn't have to be on a mammal, it will be present on the dried leaves on the ground and all. So every time the farmer goes for collecting, farmers or the rural people go in the forest for collecting firewood, even they will come outside with ticks on their clothes because they're there on the leaf. So there's a season. So now the people staying around that forest have been made aware. Like in this particular season, during this particular season where the larval reproductive cycle is going on, when the larval stage is prevalent, they have been told to use tick repellent. First of all, not to go in the forest. If they have to go, to use tick repellent. Okay. So if you go to the forest, get exposed to the wildlife there, then you get ticks on these wildlife, and it can be come to humans. So once the ticks feeds on the human blood, then it gets infected with the virus if the tick carries the virus, and then the infection gets prevalent in that region. Okay, this is the life cycle of KFT. So as I said, KFT was prevalent only in Karnataka, but now cases have been, or seropositivity have been identified to other regions or states around Karnataka. Okay, so the infection is spreading. These are the factors which the environmental scientists, researchers have identified the factors for KFT prevalence. First is deforestation and increased animal interactions of humans. So every time, now they are aware, every time there is a dead monkey, no one goes close to the dead monkey. So this was the replication cycle of KFT. So you can see, it's a positive sense RNA virus. So it is positive sense means it's like the mRNA. For us, it's like the mRNA, right? So it can directly get translated into protein. So the virus enters the cell, the genomic material enters the host cell, the RNA gets translated into the protein it requires for application, and once the protein is formed, then the protein is basically the polymerases that it requires for replication. Once that is formed, it will replicate the positive sense RNA and form new viruses. Incubation period is 3 to 8 days. It causes different symptoms from fever to neurological symptoms, that is encephalitis. So the neurological symptoms happen in the second phase of this. It's a two-phase disease. First is just fever, febrile, fever and flu-like symptoms. But after a few days, not all of them, but most of the cases, they will develop neurological disorders also. Clinical samples that we use for detection of KFD is blood, serum, and if it manifests neurological symptoms, then we collect the CSF, that is cerebral spinal fluid. These are the diagnoses available for KFD. Point of care diagnosis. What do you understand by point of care? Anyone? Point of care diagnostic devices are assays or devices which do not have to be operated in the laboratory or have to be performed in the laboratory. They can be performed in the field at the site, right? So it can be an assay which like a PCR assay where you just have to add the sample. Don't need a thermal cycler as well, just a heating block, and it's called lamp assay. And that's a POCT. So if there is a KFD infection, the rapid response team will carry these POCTs to the field, and they will test the samples of the monkeys or of the humans. They don't have to send it back to the laboratory. Treatment is these are the treatment. These are symptomatic treatment. They are not exactly antiviral treatment. They will treat the symptoms of the patient, the fever, the neurological encephalitis, and all, right? Vaccine. There was a KFD vaccine available, but after the use of almost 2 decades, they realized that this vaccine is ineffective. Now the vaccine is discontinued and new vaccine is being developed. So what are the key strategies? Vaccination, awareness program, as I said, the moment there is, and surveillance, continuous surveillance of monkeys as well as human cases. Nipah virus, this is a second example. I think we'll take another 5 minutes. It's a risk group 4 pathogens, okay? And handled in a BSL-4 laboratory. It's a negative sense RNA virus. So when I say it's a negative sense RNA virus, don't have the figure. So a negative sense, it means after the genome enters the host cell, the RNA will get transcribed to a positive sense, to a mRNA, okay? So there is transcription happening. That mRNA will again get translated to form proteins that it requires for assembling the the viron particles. So there are 2 levels, transcription and translation. So at every step, there are chances of mutations developing. So the more the procedures that it is undergoing in the host cell, the more is its potential to develop mutations. So that is why RNA viruses are more or susceptible to mutations and adaptations. So most of these outbreaks happen due to RNA viruses. That's the geographical distribution of the Nipah infection, and you can see the red dotted line that shows the range of Nipah, as I said, is originating from bat. The red dotted line shows the flight range of bats. So you can see it can cover from bats from Africa can cover and go to Asia and also reach till Australia, right? So that is how the infection spreads. So it was first identified in Malaysia, where they identified that the infection came from bats to pigs and then to humans. Then it was reported in Bangladesh in 2001, and there they said that the infection came from bat. The bats ate palm, toddy palm, or the urine of the bat entered the toddy palm sap. Now toddy palm, if you may know, the rural settings, the toddy palm is consumed as and like an alcohol drink or used for cooking and stuff like that. So it was noted that this was the route of Nipah transmission in Bangladesh. Now Bangladesh is close to India, so yes, in after Bangladesh reported Nipah, it came to West Bengal, Siliguri. Okay, after it came, 2001 it came to Siliguri, West Bengal. 2007 again it reported Nipah infection. From say West Bengal, it came to Kerala. Nowhere else it was reported. It came to Kerala in 2018, and from since 2018, Kerala has been reporting Nipah infections after every 1 or 2 years. So there is a periodicity noticed. It leaves a gap of 1 year. Okay. So, but in 2026 again, 2 cases were reported in West Bengal. Also here there is Nipah. I just explained the replication cycle. You can skip this. Incubation period 4 to 14 days. I said even within 4 days of exposure, it will the patient will start showing symptoms, and the mortality 40 to 75%. Pigs, dogs, cat, all are reservoirs for Nipah. The samples, swab, nasal and oropharyngeal swabs are used. CSF. Actually, for Nipah, all samples are taken. Oropharyngeal swabs, blood, CSF, tissue samples, neurological, brain, everything is taken for Nipah. You don't leave any loopholes when it comes to Nipah identification. Diagnosis, RT-PCR and ELISAs for serology. And ribavirin, that is again a symptomatic treatment. It is not a virus-specific treatment. Vaccination, no vaccination till now. No approved vaccines have been in the market right now. So what happens when there is a Nipah infection? The government appoints a rapid response team. The rapid response teams will have environmental researcher, clinical researcher, and medical practitioners. So that team, and they are being trained to handle BSL-4 or risk group 4 pathogens. So they are deployed to the site where they go and do investigation, outbreak investigation. They go and see which is the index case, which was the first case, whom all did they contact, how did they get the infection, where all did they have visited. So it is a very humongous task to do contact tracing. So they will take all the history of that particular case. They will ask them where did they visit, and they'll go there and talk to people who had come in contact with this individual. All their samples are collected. Field, and there is also bat surveillance. That is One Health approach. So Nipah, so bat is the source. So you monitor the infection in the bat. Okay, so even in West Bengal and even in Kerala, the bats are surveyed continuously in each season. And laboratory establishment. What is worrisome about Nipah in India is like we do not know the route of transmission in India. In Malaysia, we know it came from pigs. Bangladesh, we know it came from todi sap. But in India, it has not been identified where is it coming from. In Kerala, suddenly there'll be students going and visiting some waterfalls, and they come back and they get infected. What happened in the waterfall, we were not able to identify. Some, even now in West Bengal, 2 healthcare workers, 2 nurses got infected. How? They had visited some rural area, but in that rural area, when we did surveillance, Nipah was not identified. So how did they get infected? No one knows. So that is again a big area of research. What is this transmission route of Nipah in India? So these are all potential routes. First was todi palm, but it has been found negative. Not containing presence of Nipah. Bat consumed fruits. In Kerala, if you go in these endemic regions, they'll be scared of consuming any fruits, any of the local grown fruits. They'll be like, no, we won't. We are scared of Nipah. So, but it's not been established or proven that Nipah has come to. When you take these fruit samples, Nipah was not found, right? But people are scared. Okay, people have been made aware, and they stay away from these half-eaten fruits or fallen fruits. So what has this pandemic preparedness or all this strategies done? If you see in 2001, 2007, in the earlier Nipah outbreaks, cases were 65, deaths were 45. But if you come to 2026, 2 cases and we contain the infection. So that is how preparedness has helped to contain the infection. Before it spreads to the masses, you can go there, you do surveillance, you contain, you give them treatment, right? So these are the other emerging and re-emerging viruses. All are present in India. Some of them like virus and all, they do not cause, they have not shown to have caused severe infection in humans. They're serologically positive, means their antibodies have been found in humans, but whether they cause any infection or not, it's not known. But why are there the potential risk emerging infections? Because they belong to the Buniaviridae family, which have other viruses which are pathogenic, right? Chandipura virus, Zika virus, these are all Indian emerging and re-emerging infections. So with that, I conclude my talk. If there are any questions, please feel free to ask. No questions? Should I ask questions? And I think you will have some MCQs after this, yeah. Yeah, ma'am, because we were not prepared for such large scale infection, as simple as that. We detected SARS-CoV, by the time we detected it may be a coronaviridae family. And it came, first of all, we couldn't locate the source, that it came from a live market, it was a zoonotic infection. By the time and we didn't have the tools for diagnosis. This SARS-CoV-2 was very divergent from MERS-CoV and SARS-CoV, which are the family, which belong to the same family as COVID, it was very divergent at the genomic level. So we do not have the tool for diagnosis. By the time we did whole genome sequencing, we identified it's a coronavirus. After we identified coronavirus, WHO and other global agencies, they developed probes and primers and probes for its detection. They disseminated it into different countries. It was too late by the time it had spread. The second reason, the transmissibility of SARS-CoV-2 was very high compared to MERS and SARS-CoV. Okay, it was found. It's called the
request all of you to please settle down. We'll start with the session now. Welcome back, all of you. And today we have the second talk speaker with us. I request Dr. Meenakshi to please occupy the chair on the dais. Please come. Yeah, you can. May I request Dr. Prasanna Srinivas, HOD of Department of Microbiology, MS Ramaya College of Arts, Science and Commerce, to present a sampling to our speaker as a token of respect. Thank you, sir. So good morning to everyone. Once again, I welcome you all for the second talk of our today's lecture on advanced biomaterial technologies for tissue regeneration using 3D bioprinting by Dr. Meenakshi Kamraj. Dr. Meenakshi Kamraj is a DST-inspired faculty in the Department of Bioengineering at the Indian Institute of Science, Bangalore, and an affiliate researcher at Terasaki Institute for Biomedical Innovation, United States. She received her PhD in biomedical engineering in the year 2023 through a joint program between IIT Hyderabad and Swinburne University of Technology, Australia, where her research focused on polymer and ceramic-based bioinks and scaffolds for tissue regeneration using 3D bioprinting and advanced biofabrication techniques. Her work spans bone, cartilage, vascular, muscle, and wound tissue engineering with a particular emphasis on tissue-specific bioinks, reinforced scaffolds, microspheres, and nanofibrous systems. She has extensive international research experience, including at Biofab 3D Labs, St. Vincent Hospital, Melbourne, Saga University, Japan, and Terasaki Institute, United States. Dr. Kamraj has published extensively in leading QN journals, received several prestigious awards, including the DST-inspired Faculty Fellowship, Supra Fellowship, and the IIT Hyderabad Research Excellence Award, and actively contributed to interdisciplinary research, mentoring, and peer review in biomaterials and biofabrication. Madam, we are very much pleased and privileged to have you, your lecture in our MSRC-AC KSTA jointly organized lecture series. I thank you on the behalf of MSRC-AC and KSTA for your acceptance to be part of our program. Thank you. I wish you all to have a great day ahead. Now the session is over.
Thank you. Thank you, Professor, for a generous, brief introduction. So, hello everyone, I'm Meenakshi. As he already mentioned, I'm a DST-inspired faculty in bioengineering and Department of Bioengineering in IISc. So, today it's going to be a kind of different what you have been hearing from past 2 days. So, I guess you guys must be listening for a lot of basic sciences and application of basic sciences in health, or maybe a basic research on understanding disease, but then this is going to be a completely different one. So this is how we approach using an engineering perspective to a medicinal or healthcare system. So how many of you have quite some interest in engineering? Very few. So I think all of you are going to get bored in my talk, because it's going to be quite a lot of engineering with application in, tissues or organs or regeneration. But I'll try to keep you engaged. I don't want to be talking from a single end. I want you guys to be, interactive over the presentation, so that you guys will know what's happening and what's research in bioengineering we do with different engineering perspectives on it. So this is where I have completed my PhD. So I completed my PhD in IIT Hyderabad, moved to Swinburne, and then I worked in St. Vincent Hospital. The reason I'm showing this, so this is what is shaping my future. So I also from my BTech is from a, a state college, and then I moved on to a Pondicherry Central University. Then I moved on to IIT Hyderabad. So it's not about where you come from. You need to have a passion for that, and you have to have a hard work for that. So that's why you can even accomplish what you wanted to accomplish.
So these are the things it's still, shaping my career, and then how I see a problem and how I approach the problem in healthcare field. Because as I mentioned, so this St. Vincent Hospital has a combination of 8 institutes, which is academic institute and also a clinical institutes. So what we do is see a problem in a clinical and try to address them using our engineering approaches. So it is not a single department job. It's an interdisciplinary job between different institutes and research labs to focus on some particular problem. And then I moved on for my postdoc to Los Angeles. And how many of you know Los Angeles is famous for what? I guess you guys might have heard Los Angeles. So what is this famous for? I can't hear anything. Be loud, no? I am loud enough for 100 people. Can't you people loud enough for one person? So what is this famous for? Hollywood. But then it's not about Hollywood and then your Universal Studio Hollywood. It is also famous for biotech startups. So all the biotech startups emerge from Los Angeles and San Francisco. So if you go Google search it, how many biotech startups are there, you will find a hell of a lot of them. So it's going to be a lot. So all these biotech companies, vaccine producing companies, and then tissue regeneration companies, all these startups are emerging from Los Angeles and San Francisco. So that is one of the most famous part of Los Angeles. So my institute, I also worked in Terasaki, is a non-profit research organization. It's not a typical university where I work. So our idea is to, have a biological problem, but then how we patent them, how we make them, have a product over time. It's not about you do basic research, but then you keep on doing basic research over the time. It's like how we can make them functional over the time. Of course, medical, implants or scaffolds or whatever we have it takes over a long period of time, 10, 15 years to come to clinical market. But then what we wanted to see is how we can start from now, like TRL1, how we see the problem from TRL1 to TRL8 to see how we can grow over the clinical product. So that's what my research organization was doing, and still they are doing in Los Angeles. And it's apart from that, this institute also provides, lot of research opportunities, and then, we also provide this community-based outreach program. So what we usually do, we call people from outside of the institutes and neighborhood people, and then call them for a research, You do a fancy, very sci-fi research, but then if you are not able to, summarize them or explain to them to a 5-year kid or something, you are failed to do your research. So whatever you do, it needs to be translatable easily to a layman language. So that's what we usually do. So every year we also organize some community-based research program, and then we do every summer, winter researches for bachelor and some master students, so that they get experience of it, what we are doing in a biomedical innovation. So that's our idea of working, how you start from clinical approaches, from where I started my PhD, working on clinical approaches. Then I moved on to a place where we see them in a product-based development and having an outreach programs. So that's what it's still shaping my career, and that's how I am trying to see how I can explain to people in a very basic level. So that's how it's going to be today's talk, and I hope you will be interactive over the period of 1 hour, I could say. So this is my work experience, so in tissue regeneration. So I'll come to what is tissue regeneration and engineering next slide, but this is just the experience of my research experience in tissue regeneration using different polymers. So people who are in chemistry or related to quite biotechnology might know what are these polymers. So I have a quite combination of polymers like natural, synthetic and semi-synthetic polymers, and then have them in different methodology or techniques has been used for making these polymers into a scaffold system, and then use them for different engineering applications to address some tissue, dysfunction or failure. That is mostly talk about, vascular and skeletal muscle and bone and wound and cartilage. So I have quite wide experience in fabrication techniques and then also in different tissues. But then for the talk today, I'm trying to narrow it down to musculoskeletal, where I'm going to talk about bone and cartilage. So I'm not going to brief you everything, so it will be, above your head. It's too much to take on for a single time. So we will go ahead with only today for bone and cartilage. So what is tissue engineering? Anyone? What is tissue engineering? Any? It's need not to be a correct answer. I'm not expecting a very perfect theory book answer. I'm just expecting what is tissue engineering from your understanding. So what's tissue engineering? Yeah, similar. So what we try to do is we try to repair them, and then or else we try to replace them, and or else we try to regenerate them, right? So how many of you watch Marvel comics? Very few Marvel people? Really? I see only 3 hands, 4 hands, then I slowly see few of them are like, nobody watches Iron Man, nobody likes Captain America. Really? Really? You guys are Gen Z kids. Don't tell me that you don't like anything. Okay? Or maybe you guys are ahead of Marvel comics then. Only I stuck in Marvel comics then, right? So, what's this? So what's the first one? Okay, then what's the second one? What is the second one? Very, very low voice. I can't even hear. All I hear is like a hiss. This is what I'm hearing. Yeah, the Avengers headquarters. And what is this guy? Nick Fury. And then what is this? So this is the Tony Stark's garage where he built all his Iron Man suits and then helped to make, the Thor and then Captain America instruments, right? So this is a similar thing what we do in tissue regeneration. So what we have, these superheroes as your cells. So that is what is there in your body. But then more I talk about stem cells, because these are the stem cells which we could, differentiate into them a different organ or tissue. That's why I call these are the superheroes. And then you have, this is the place there, Tony Stark is a, so this one is basically your scaffold. So all these materials, all these superheroes assemble in the same place, right? That's the same way. This is the scaffold. It's all being encapsulated or being there in a single place. And then Nick Fury, what is the role of Nick Fury in Marvel? What's the role? Huh? Really, you guys don't watch Marvel? Or am I missing something else? So what does he do? Really? Yeah, same thing. So he basically tells them what to do, right? What is your next action of work? So that's the same thing. So you have your cells, you have the place where they assemble, which is your scaffold, but then you have to make them work, right? So you need them have a communication to do what they wanted to do. So that is where your biomolecules or growth factors come in. And then we have the place where they actually make things. So that is where we fabricate things. So whatever I talk about fabrication is like a garage. We basically make different techniques to make them fabricate some different scaffold for tissue engineering application. So this is a simple term of how we correlate. So basically how you correlate your basic science into a very small layman language is important. So if you tell this to a 5-year-old kid, even the 5-year-old kid will understand what is tissue engineering. I hope you will not forget what is tissue engineering, right? So what is tissue engineering now then? What's tissue engineering? Yes, with the help of scaffolds to repair or regenerate an organ function. So that is our tissue engineering or tissue regeneration in terms of when you talk about repairing an organ. But then tissue engineering falls into different categories. So the previous speaker talked about point of care. So what is point of care? What is POC? Yeah, so it's like a glucose monitoring kit. Everybody have at home, right? So that's application of tissue engineering. So it's an engineering approach basically. It's a basic biological thing, but then it has an engineering approach. How you emerge them from a laboratory to a normal person, how you can utilize them. So that is your tissue engineering. So tissue engineering has like a broad spectrum. What you approach, it's like you wanted to have a disease, diagnosis. So point of care is a diagnosis, or else the other part is where you wanted to do disease modeling. let's say somebody has an organ failure, let's say it's a renal kidney organ failure. You know it's happening, but then you don't understand the whole pathway of it. So tissue engineering has an approach to do disease modeling. That is what we use it for disease control. And then the other one is the repair and regeneration. That's what I am talking about, is tissue regeneration. So it uses the engineering approach, but then what I am going to talk about is regeneration, or replacing an organ function is regeneration in the category of tissue engineering. So this is a basic, supposed to be, this is the first slide, but I know this will be too much for you guys to take what is tissue engineering. So this uses, tissue engineering uses the building blocks of things, which is your scaffold, which is polymer and cells, where I talked about superheroes and the where they assemble, and then you use them to make them in a different forms. So which is, you use different fabrication forms. So whenever I talk about scaffold, it has some shape, okay? This is something you have a shape them. So that is all these scaffolds come in. With cells or without cells is depend upon which, how we approach or use them for. And then we use different type of techniques, but mostly today I'll be sticking on to additive manufacturing. So what is additive manufacturing? I heard some people have, engineering interest, but what is additive manufacturing? So it is a basic 3D printing. Additive manufacturing is anything you do in terms of 3D printing. It could be any different type of 3D printing. There are varied types of 3D printing available. It depends upon, again, which type of material you use it and what structure you wanted to fabricate, but additive manufacturing is basically a 3D printing technique. So that's what I'm going to talk about today. So what is 3D printing? What's 3D printing? Huh? I don't hear anything. You guys had a coffee break, right? Yes or no? Yes? Say loud it. So then you should be active, right? There is some sugar molecule. You have energy to talk. I didn't even have a sugar molecule. I'm like already drained. So be loud. So what is 3D printing? What is 3D printing? I'm not going to assess your knowledge. I'm not going to give you mark. Don't be so shy. What's that? So basically, this is 2D printing. What you do taking a printout is basically a 2D. You have something, you just give it to a Xerox machine and then you get it done is your 2D. But then what I wanted to do is 3D printing, right? I wanted to have them in a 3-dimensional range. This is in a 2-dimensional range of X and Y. I wanted to do in a Z direction too, in a 3D protrusion. So that is what we do in 3D bioprinting and 3D printing. So we wanted to make them something from 2D to 3D. I can fabricate them. But then again, there are different types of 3D printing. I'm not going to talk about all different types of 3D printing. All I could talk about is 3D bioprinting. That's where my work basically is on. So then mostly we talk about today is 3D bioprinting, but this is basically a 3D printing. So this is like a filament-based approach. You see there is a line going here on top of it. So this is where the filament has been, fed into the printer and it melts and then it makes your 3D model. All you need to give is the file of what you wanted to print. So you give Eiffel Tower and it's going to print your Eiffel Tower in quite some time. So that is what is your 3D printing. So then I talk about 3D bioprinting. You can make 3D scaffolds, which is in a 3-dimensional X, Y, Z. But then when I talk about bioprinting, there is going to be some other polymers or cells involved in it. That's why I call it bio. When there is bio coming, your cells are involved in that. So basically these cells are stem cells because I talk about regeneration, right? So that's where you have stem cells involved in it so that they do a function of repairing or regeneration of the organ. So we have polymer, we have cells, which we already saw. Your superhero and then your polymer is the place they assemble, and they have bioactive molecule, which is a Nick Fury. And then what you have to do is go to the garage and make your scaffold. So that's what we do. We make a 3D bioprinting and then we do a 3D bioprinting of different scaffolds for different organs. But then it's a typical theoretical approach of what we call is a layer by layer printing, because it's going from one layer to other layer to print a 3D structure in Z direction. And then you can control and functional architecture. You will be able to control your like line width, and then how you wanted them to print, and what is the size you wanted to print, and how it could be. So this all you could control them. And it's basically called a bottom-up approach because we start from base and it goes up. So it starts from Z level in 0 and go to the Z axis till the size you wanted, let's say 10 or 15 centimeter. That's what is in Z direction. So this is a typical 3D printer, and then what you make some 3D scaffold. I think it's the ear cartilage is being printed. And what we do, there are multiple parameters affects it. Like it's too much from the materials, but I just try to brief you what it affects it. So one I talk about is printability and shear thinning. So when you use the paste, everybody brushes in the morning, right? Right? Or no, you guys don't brush it. So when you press the brush, the paste, your paste comes out, right? So that's shear thinning. So when you press, put a load on it, they extrude. That's what extrude is terms of technical term. That is coming, like probably it's like thinning is extrudability. Shear thinning is when you apply load, it's going to be extruded. But then you kept it at a load 0, where there is no pressure. What happens? It is in a resting condition, right? You don't see them coming out of your paste. So that is basically a shear thinning property. And that is what we call also printability, because what you do is a shear thinning, but then when I wanted to put them in a 3D printer and then print like this is called printability. So this is what is your printability. And then structural stability and degradation. It's structural stability. Let's say I wanted to print something in 4 or 5 centimeter in a 3D printer. It could be basically 1 to 2 centimeter, but then I wanted to make something in 3 4 centimeter, then it has to hold the structure. I cannot use the paste to make it 3 4 centimeter, right? So I need to see how good the material is, how good it could make the structural stability of the scaffold and all. So that's one of the parameter we talk. And then the next one is biodegradation. So you guys might know degradation and biodegradation by enzymatics. So how many of you are biotechnology and some basic biology courses you have done? So how does degradation happens in body? Same thing. So it's the same thing what happens in biodegradation. So when you put some material inside your body, how it is going to react with your cells in it, and then there is going to be degradation. That's called your biodegradation through an enzymatic mediated degradation process. So that is also we wanted to see how it happens over the time, because we make the scaffold and then we put them in the human body, or let's say in animal, but then what happens over time? Are they going to degrade? How fast they are going to degrade? That is also important, because let's say I made a bone scaffold in a polymeric form and then I put them inside the human or animal for a clinical trial, and then I find out that it degraded in 2 weeks. Is it enough? Is it enough for bone scaffold to be there for 2 weeks? What happens if it goes in 2 weeks? If it's like degraded in 2 weeks, what happens? Then, what's the other thing? Yes, and then? Yes, that's what I'm expecting for. So, it needs to provide a structural stability or a structural strength, right? Bone is the hardest tissue in your organ system. It holds the pressure or load in gigapascal. We talk about something in kilopascal or pascals. So your load or pressure, whatever you put, is very less. I press with my thumb, it is in few pascals. But then what your bone and then your femur or something in a knee takes it in gigapascal, the whole body. You jump, it needs to withstand the load. You walk, it needs to withstand the load. The similar way, I cannot put a bone scaffold with a lower mechanical strength and expect it to regenerate bone again, right? It needs to have a similar properties of it to regenerate. So that's why we talk about how good that material could be and what happens to them over a biodegradation process. Are they going to degrade faster, or are they going to stay over a longer time? That's all come under your biodegradation. And biocompatible and aid in repair is basically how these scaffolds react with your cells in the body. Like if you put them, or is there going to be immunological reactions? So I see few people in the back, some bio. Maybe you guys know what are the cells in immune cells in body? And then, yes, Yes, so most of the time what happens is all these immune cells will come into contact when you implant implant into the body. But then what tunes them into repair and repair or regeneration is your macrophages. So what these macrophages will do, there are two different types already, M1 and M2, which is, what is the M1 and what is M2? What is M1 macrophage and what is M2 macrophage? So it's pro-inflammatory and anti-inflammatory, right? So M1 is your pro-inflammatory macrophage, which will come and attack your material, and then the second one comes in where it will differentiate from M1 to M2. Let's say if they differentiate from M1 macrophage to M2, this is where your regeneration happens. But let's say if they fail to repair, move from M1 to M2, what will happen? They will keep going in M1 phase, which is your inflammation phase, where the continuous inflammation happens. So that is called your chronic infection. So that is where this macrophages play a role. That is also we wanted to consider when we talk about biocompatibility of the material, right? So that is one of the important parameter. All of these things, it's going to affect your repair or regeneration process. That is what we talk about in tissue regeneration, and these parameters are going to affect your 3D printing and the scaffold and the repairing process. So this is a basic thing. What we do is you have a source of material, it could be any of the polymer or anything, and then you have a 3D printing and you fabricate any type of organ or tissue, and then you do a in vitro studies, basically a lab studies on in vitro level, and then you make them use mature them and then use it for implantation or translation into a human application. That's what we do in terms of 3D bioprinting for tissue regeneration. So the first one I'm going to talk about is on polymers for bone repair. So it's how we see a bone. Too many people, we know elder people have osteoarthritis. So we know this is a clinical challenge in India. But then how we approach them is what we are going to see now. So bone consists of, one of the major thing is in bone is higher concentration. What is that? Calcium. So what we have is highest calcium phosphate, which is hydroxyapatite, HAP, that's what we call it in a chemical term. And then we have some water, and there is some organic parts. We also have some collagen. If you see, the collagen content in bone is also around 20%. So we wanted to have something of this. But what we do in clinical now is mostly when people have some, trauma or something, what they usually do is metal implant. Basically to give a support. That is where they use for secondary support. This is not your primary care. This is basically you use them to support your structure so that it provides load transmission. But then what happens here is you have a metal. Metal is just, it's not going to degrade faster. It's nothing going to happen to your metal over the time. So what they do is secondary surgery. Over the time, maybe the bone ingrowth has happened, but then your metal needs to be removed because now it is affecting your movement. So what they wanted to do, they will go for a secondary surgery to remove your screws or bolts has been kept in your bone. So that is happened over this cause of trauma or some bone accidents. But then what we wanted to do is how we avoid metal. We don't want to use metal because metal is something, most of them are bio-inert, it provides some support, but some of them have an osteoinductive property to, osteoinductive in terms of osteo is your bone cells, inductive property to help them repair. But then how we eliminate the metal and use something scaffold which support the growth and also which provides the, the mechanical loading condition what we expect for them to do. So that's where we come talking about 3D bioprinting for bone scaffolds. So my first project was mostly on 3D bioprinting of different types of bone scaffold, starting from some polymer materials and then using some ceramic materials and then using ceramic and polymer together to see how these scaffolds will work over the time. So starting from the first one is functionalization of photo-cross-linkable polymer. So the name refers photo-cross-linkable. So what does that mean? What is photo-cross-linkable? Its name suggests photo-cross-linkable. Yes. So something, it has been interacts with light. How many of you do nail art and go for nail, nail designs? What do they do? What is your extension nails are? Huh? What is your extension nail? How many of you go for nail art and nail designs? No one? Are you guys are too shy, don't want to show the other people because they will go to the same place or something? Is it something like that? Huh? So what do they use there? Your extension nails are what? Huh? It's a polymer, right? It's a photoresin. What they put in your nail and then what they show is your blue light spectrum, which is UV light, and then make them cross-link. What happens? There is a cross-linking happens. That is what I'm talking about is photo cross-linking. So the similar thing is happening here. It's being cross-linked, but what all you do is a nail, but then what we do is bone. That's what I'm talking about. So then we have, so I had one of like, during my PhD, we had one of the chemistry postdoc. She did her PhD in chemistry. So we had a, very good chemistry postdoc in a lab so that she do all our chemistry synthesis, and then all these engineering biology people, what we do is just borrow her material and do all the 3D printing and then all the fancy stuff and then use it. That's what happens. But then it's again, talk about when it comes to basic, you needs to know what you wanted to prepare and you are good at doing it. So she has a very, passion to do what are the materials she could do. Back in 8-9 years, photo cross-linkable materials are not very fancy. Now you all talk about nail art and everything, it comes very, common. But then back that time, it's all in research. What we do is synthesis in your own lab and then make them for some application. But then now we can able to buy these photo cross-linkable materials very easily. That's what happened. But then we like synthesize a lot of these photo cross-linkable materials. I say PEG is called polyethylene glycol, and alginate is a seaweed derived material. It's also biological, like it's a biopolymer. And then we do some functional group, which is your photo cross-linkable group. That is where your all these blue light cross-linkable or UV cross-linkable thing comes, which is acrylate or methacrylate. The name suggests acrylate is photo cross-linkable moiety, which is being attached. So what happens is this acrylate moiety being attached to your PEG, and then which makes these network after cross-linking. So this network is now entangled that after cross-linking they don't move. That's what happens in these materials. So we print all of them with this material with your cells and with the photo initiator, because when there is photo cross-linker, they use it, they need to mix certain materials in a certain ratio. Let's say you have only some material, which is, let's say my PEG, I put it there and then let them expect to, start the reaction to cross-link, they won't cross-link. Because you need to have some initiator molecule, like a Nick Fury, who has to tell what need to be done, right? The same way, they will do that and then they will make a cross-linking and then they make a complete structural scaffold. So this is what we try to do is like make a different combination of polymers. So we did all of them with the different acrylate or methacrylate groups. And then we see that it is blue light in the sense, you shine a blue light in the range of 395 to 405 nanometer, they cross-link. And then we also cross-link them with calcium chloride, because I said there is something called alginate is here. So alginate, I mentioned, is a seaweed derived polymer. So it could be cross-linked in different mechanism. So what happens is, if you have something divalent ion, it will cross-link. So if you put anything like calcium chloride or magnesium chloride, you put any chloride which is divalent, Cl2, 2+, it will cross-link. But then if I put something in single valent ion or monovalent ion, what will happen? It's a reversible reaction. It will keep on going between reversible. So it will again leach out and it will become gel again. It's no more solid. So that's what happened with this, the alginate group. So what we tried is we cross-link them with calcium chloride so that it make a stable structure. And when I mix both of them together, I need to cross-link them in two different conditions, right? I cannot just cross-link with blue light and expect the other material to stay. So I have to cross-link with the other solvent also to cross-link them in two conditions. So what we do is we make them and we cross-link them in two different conditions and study their properties over the time. So first I talk about 3D printing. So whenever we make some material, first thing we try is see that material is first photo cross-linkable, and then we tried photo cross-linking and it's working. The second thing is to see if they are printable. So So this is where we talk about 3D printing. So when you see there's a very small droplet on this, so this is where there is a droplet formation. That means that it is not 3D printable. All it does is like when the nurse take the needle and then they check the water comes out or your drug comes out, it's like the similar thing. What I see is just a water droplet is coming, nothing else is coming. But then what we wanted to see is basically this filament structure. A continuous source of filament needs to come out. That is where it is printable. Okay, there is a filament comes in, so that over the time when we 3D print, there is no disturbance or breakage between the filament length or width. That is what we expect. So then we 3D print them, and then this is before, like it's after 3D printing, but before cross-linking. And then the next are like after cross-linking. So you see like a visible change, right? So here it's something transparent to here you see something translucent or opaque. So it's just because there is a cross-linking happens, and that cross-linking make them translucent or opaque. That is what happened. And then once it is cross-linked, we will be able to handle these structures. So that means you are able to cross-link them and you are able to print them and then handle in your normal conditions. So the next we have a polymer. We then made them into a scaffold, but then we also wanted to talk about regeneration. So this is where your cells come in. So in this project what we did is, it's for bone, so we wanted to have something differentiate into bone. So how many of you heard of mesenchymal stem cells? So these mesenchymal stem cells could differentiate into different, what different? Yes, so it could differentiate into different organs. So this mesenchymal stem cell can differentiate into cartilage, bone or adipose, or sometimes if you give a proper cues, say growth factor or biochemical cues, they will also regenerate or like differentiate into like vascular cells or vascular smooth muscle cells type of thing. So that's what we wanted to do, because isolating from bone is going to be very difficult, challenging. Like expecting a 60-year-old to have a active bone cells is not easy. They will not have something active bone cells there. So we cannot have them expected them to be like isolate and then put them in these scaffolds is not possible. But what all we could do is having some stem cells, which is mesenchymal source is very easily available for us, and then we put these cells inside the material and then 3D print them and then see if they were able to differentiate into different organs. So we tried to do this live death staining. Basically it's a biological staining. It's a biofluorescent, I would say it's a fluorescent assay. You put calcium AM, which like transfer to cell membrane inside the nucleus, and then it expresses green color. That's why you see green. So where there is a green color cells, it's all live, and where you see red color cell, it's all dead. So basically when they penetrate inside the cell, they will detect into some enzyme, and which fluorescent red basically is dead, and then in green it's live. So that's what we usually do, see what is live and what is dead after the 3D printing, because there are a lot of parameters is going to affect. One is your material, one is your 3D printing, because you are going to 3D print them. It's going to take over some time. The time is also matters. Your cells cannot be live in a in vitro condition outside of his normal mimicking condition to be alive for some time. So we also wanted to see how they will react over time. So this is what first thing we do, talk about biocompatibility. And the next one is to see how they actually proliferate over time. These cells are proliferative, but the proliferation times are different. For mesenchymal stem cells is very less. But then when we talk about cancer are very highly proliferative. So cancer is like very, they say that its recurrence happen over the time. It's very fast just because they proliferate faster. But then in case of stem cell, the proliferation are very less. But then we wanted to see, are they able to even proliferate over time in our scaffolds? So we do some of these assays. It's called Alamar blue assay, or like dye. It's basically a dye reduction assay. Similar thing. When you use these dyes, what happens? It's like, have you guys done a protein assay, BCA? Some biological, biotechnology courses, have you done some BCA? So what happens there? What you measure? So what do you measure in BCA? Or what do you measure in any absorbance study? what do you see is a color change, right? Any assay you do, biological, biochemical, or any assay, or even you do ELISA, what you see is some time of incubation and there is a color reaction happens. So that is what we do. So there is a color reaction is going to happen in this assay. So what happens is that this reduction is basically a color reduction. So what is that? Is the color reduction is correlating your cell survival or proliferation over there. So that is what we do. So we try put these colors into the scaffold and then we let the chemical or the dye to sit over time for 2-3 hours and then we see what happens over time. And then there is a color reduction. What happens is it's a blue color dye and when you put and then the cells start reducing it by its enzymatic reduction. So active cells will reduce. The cells they are die, they are not going to reduce because they don't have an active enzyme. So that's the same thing. When they have a higher reduction, that means higher proliferation. That's what we see here. So there is a high reduction in this sample. That means this cells are highly proliferated in that sample. And then also we see as we talk about this mechanical and for some more sample, we also wanted to see how they are stable. How mechanical, like which percentage of Young's modulus is basically to measure how much load you can take. So we try to see how much load you can take samples and we found that. The next thing what we found is all different things. So we try to think, then we talk about biocompatibility, which is saying what as a shape. So basically you have then you wanted to make a certificate. Let's say that these are things you see, right? Either you do to protein level. These are more specific marks. You know, formation happens because happening. And then next two is a molecule transcription. Cells and says that you move faster and then the matrix it needs to be stiffer. Talk about because there is a like contact expression level and then other thing so we want to see what is the there are other things that is easy absorb other things are absorbed. So first you do a protein expression which is so the we can talk about RT-PCR and ELISA, right? So what they do is to find the diagnosis. But what we do to see there is expression of some marker present in there for some application. So that we did for some and then Saranax 2 and 4. This is all three marks we talk about here for analysis of bone specific expression. So we quite done an RT-PCR study to see which of the material has higher expression over time. So these are find that we could be able to see that there is some higher expression happens, but then we see in the second one, which is PEG related, and then A is algae related, to see there is some differentiation happens in these scaffolds into bone specific pH or specific iron type. So next we see this PCR is done, which is the gene expression level, and also we wanted to see protein level translation happens. So there is transcription happens, right? We wanted to see this translation happens in the protein level. So we go to that and see there is a ALT expression. So we do see a ALT expression for all of these samples over different time, because it's not you make a sample and you do not expect them to express all of them in 1-2 days. These expression takes over weeks. So these studies are like done in this is day 14 and this is day 28. Basically it's like day 7 and 8, day 7 and day 14 or day 21 or day 28. It happens over the month. So we let them be there over in vitro studies to see are they able to differentiate or mature over time. So we try for different time points and different conditions to see are they able to differentiate. So in this case we were able to see there are certain groups at certain time we are able to do these what you want them to do in a differentiation process. That's what we see. And then we also see that alizarin red. So alizarin red, the name suggests it is red. That's why you see a lot of red here. It's completely red. So what is this basically is that when you have a calcium, so if there is a cells which converts from mesenchymal stem cells to a bone specific cells, what you want them to have is calcium deposition or secretion by them, right? So when they start secreting calcium, so you want to identify them what like are they secreting over time. So that is what is alizarin. So this alizarin goes and binds calcium produced by your cells. So whenever you see this, so the background noise is kind of red. That's because your scaffold takes it. But when you see a dark points of red, that is where your calcium deposits are there. So these suggest that these cells are able to differentiate into bone specific cells and also able to secrete their own calcium matrix. So then it suggests that these scaffolds are able to be used as a bone specific scaffolds. And the name said I talked about 3D bioprinting. So when you talk about 3D bioprinting, 3D printing is that whenever you want to, let's say, print a patient specific or an organ or defect specific, you will be able to do that, right? Because let's say they do CT, MRI scans. What they get in CT, MRI scans? What do they do? So what you see in CT and MRI? Are you guys sleeping? Even I'm like shouting here at a high decibel, but still able to sleep. So what happens in the what you see in CT, MRI? So it's basically you see the structure of organ. So let's say there is a break of bone, you will be able to see them, right? So let's say you fit them into some modeling, simulation modeling or simulation modeling with AI or ML. What happens is that they are going to perfectly tell what is the size of the break and what is missing exactly. So when you are able to get the specific dimensions of it, what you could do is you fed them for the modeling and then you make your STL file. STL file in the which is like an fury. Either you give a biological cues, which is your growth factor, or you have some other cues which will also help them to do, which is also your mechanotransduction or material property. Your material property also is one of the cue which tell the cells to go from this lineage to a specific other lineage. So that also we found in this study. But then all of these are photocross linkable. There is something missing in this one. I'm not going to too technical to ask what is missing in, ALP or RSRL. There is something missing in this. What is missing there? So if you go back here, so what we have here on this B, so this B first one, what do we have here? Yes, and then we have cells. And what is missing here? There is something is missing. It's not bone specific. So what is the highest percentage of what? So what is missing here? So these are photocombustible polymer. They are nowhere to your bone, right? So basically, biomimetic or mimetic. Mimetic means what? Having a similar type of structure, right? That is not your mimetic. So biomimetic in a sense, it's biological system, biological organ. But then here, what we lack is something related to bone mimetic material. So that is one of the things we found. Okay, it's happening over the time. It's happening with some cues, with the stiffness or some growth factor you give it. But then there is something missing over time. So that is where we come up with the second one. Is biomimetic composite. So in this biomimetic composite, what I did is basically having, again, gel mask is a red color one, is a cross linkable. So when you shine it in a specific spectrum, it is going to cross link faster. And then you have a rate, which is, I said, is jelly. All you guys have had jelly, right? Jelly, it's all this alginate. This guy gives a better, strength to the scaffold. And then we have something called, these dots are M and O, BCT. BCT suggests biphasic calcium phosphate. So we have calcium and phosphate, both of them together in the scaffold. So then we make all three of this together. So you have some biomimetic gel. So which is photocross-linkable and also has bone mimetic, because I have calcium and phosphate, which is present in bone. Yes or no? Is it present in bone or not? Same. So we have them in bone, and then you use them with your stem cells, which is your mesenchymal stem cells, and then you 3D print them and then study their regeneration capacity again. Different differentiation studies we do, which is similar to your gene expression analysis and then your protein expression analysis. So this is similar thing, and then we modify the component to see it could have representation in these scaffolds without any cues, without any external cues present in this. So we first thing we want to see is that we did add some this calcium phosphate, but do we see this calcium phosphate? We wanted to see, let's say you mix few chemicals in the all you do is some chemistry lab or some lab, you mix all different chemicals and then you find what is happening. The similar thing, I mixed all of three, but then I wanted to know, right, after making 3D printing also I see them there, present there. So the one study we do is, first we did a SEM to see there is particle present in it, because these all are nanoparticles. Are these particles are present in it? We wanted to see some particles. And then we also do some EDS analysis, which is elemental mapping, to see these elements are present in these scaffolds. So we did an elemental mapping to see there is phosphate peak, there is also a calcium peak, and there is manganese, because there is some different type of doping we have done it. I'm not going into what doping and why doping, but then what we found is that calcium and phosphate is there. So suggesting that these scaffolds has whatever I mix together, it's already present in it even after post-processing condition. So we found that. But then we found one more interesting thing in these scaffolds. So when we tried their mechanical strength, you see the scaffold without ceramic and then the with ceramic, which is your calcium phosphate, has a higher mechanical strength. So what happens in that, when you have some nanoparticle or microparticle system in it, it actually increases its mechanical strength. So that's what we found, because it acts like some fillers. So where is the space gap, there it acts like a filler to increase their strength. So we found that it is also able to provide better mechanical strength to comparative our just a material without having all these calcium phosphate thing. So the next similar thing, we do 3D printing. We try 3D printing of some file, and then we see how the cells were distributed over the 3D printing. As I said already, all these green cells are your live cells. So after 3D printing, we were able to see these are live. And then we do see interesting stuff here is, if you see there are these cells has some morphology. It's not round anymore, right? Are these round? Do you see them here all round? Is it round? Is it at the end it looks like round, or it has some different shape? So it has some different shape compared to our previous study. So if you see my previous study, all you see was round cells, right? So the normal cells are not round. The cells has some morphology, right? They try to attach to your collagen, and then they start having some different morphology. So that is what we also see here. Basically, the cytoskeleton started attaching to your scaffold and started proliferating over time. Like they adhere, and after that they proliferate. So what happens is there is an adhesion happens. So that's what we found in these scaffolds. Because of the specific material present in it, it helped them to attach and then grow. So we see an attachment here. And then the similar thing, now we see the attachment, these cells were able to attach, and then wanted to see there is a proliferation happens. So this is the same study I talk about, there is a reduction happens. So we put some basic chemicals into it, and then we let them sit for time, and then reduce by enzymatic reaction, see a color change over time, and those color change you measure in a absorbance. So you a particular absorbance, you read them and see there is a percentage of absorbance, and then you correlate them with percentage of proliferation. So there is a percentage of proliferation, and then your reduction is correlating each other in this case. But then the next similar thing we talked about, which is having them in a gene expression level and protein level. So these are the protein level studies where we talk about alkaline phosphatase and then alizarin red. These both are staining images, shows that these cells were positive for that. And also we studied there how much is stained in this area. So how many of you are aware of ImageJ? Do you guys know ImageJ software? So ImageJ is a software where you could do a lot of imaging techniques, like measuring the specific, the stained area, and then you count a number of cells. Let's say you have two different images in two different colors in the same plane, but then you merge them together. These could be done in ImageJ software. So that's a scientific imaging software. We use it to see how much has been stained over the time, and then we measured that in ImageJ software. So I measured that and then found that the sample which is here in a pink with your ceramic has a higher expression on these markers like alkaline and then the alizarin red. So that's what is being modified, like we could see here in this graph also with the staining and then the graph, because in the staining is the stainings are very qualitative. You see and then you tell them it's okay, but then you cannot say them in quantitative, right? So that's where we do these all graphs on quantitative to see how much has been produced and how much is being reduced. That is what we see in these graphs. And then the next one is gene expression study. So we tried some gene expression studies on different markers, and then we found that the pink one actually with your ceramic were able to give better differentiation of cells, suggesting that we have a calcium phosphate. These calcium phosphate are providing a cues to cells so that these mesenchymal stem cells were able to differentiate into your bone specific cells. So that's what we were able to see in this case. And we also found out some pathway here. So we see that this happens, the differentiation happens, but then what is the mechanism? So there should be some mechanism, right? So this is the mechanism happens. So this is mostly done by ERK pathway, where there is actually a reaction where I talked about RUNX2, which is the early transcriptor guy which comes in role and then tells the cells that okay, you differentiate into bone. That is what happens. So they come in play, and where it's because of mostly mediated by your calcium and phosphate in the scaffold itself. So these scaffolds itself give some cues and make RUNX2 produce at the early, and then this RUNX2 goes over and then produce them bone-specific markers and then make them differentiate into bone-specific lineage. So the advantage of this study is basically this one doesn't have any biochemical cue which is externally applied. So the cells need some cues like someone like Nick Fury to go and tell. So there is supposed to be something biochemical like growth factor or some biomolecule needs to be present. But in this case, we don't use any biomolecule because making them or keeping them, these biomolecules are very challenging and it's very cost effective. So that's why we talk about material perspective. Making an hydroxyapatite is way easier than having some growth factor in it. So that's why we have it. That is a major advantage of it when you reduce the biomolecule use and then you use a material approach to use it. So this is purely a material based approach to make them differentiate into a different organ. So this is the advantage of having a material specific scaffold. So from here I'll move on to a second one, but if you have on first part, I'll be happy to take now. Anyone, any questions on related to bone specific one? Then we will move on to the second one, which is cartilage. No? So these cells were kept it in a very higher concentration, like 1 million cells per ml of solution. So we keep it in a higher so that these cells, even 60% of them survive after the 3D printing. That is enough to produce the remaining, because it proliferates over time. So then they will be able to take up space and proliferate. So we keep it like 1 million at least. So for these scaffolds, it's like 14 and 21. But then for some of the studies I talk about now in cartilages are like 28 days, because the normal differentiation itself is 28 days. We don't see much of the expression of anything in an early stage. We have to go till the late stage to see if that has happened. But it depends upon which type of tissue and which when these markers come in. Like the gene expression probably comes in later stage, so we'll try to find is what is the early marker and what is the late marker. We correlate between both of them. So these are mostly a BSL2 cells. So we actually, during my PhD, we had a collaboration with hospital NIMS. You might have heard Nizam's Institute. Yeah. So we had a hospital collaboration there. We usually get samples and then we isolate some stem cells out of it to have them in BSL2 can. No, so this mesenchymal stem cells is mostly umbilical cord or adipose. So it's easy to get adipose samples. Lot of people are very, obsessed with health and they wanted to have liposuction done. So we usually get some liposuction. Yeah, same. We put it in a CO2 incubator with 37 degrees Celsius, which is same as your human body. And then we also try to give these the cues which is present in the body, like the nutrient and then the CO2 percentage and then the O2 percentage, as we try to mimic on the same on a body level to keep them there. It does happen. So when we use it biomaterials, some of the materials are prone to contaminate faster. These alginate and gelatin are very easy to get a bacterial contamination happen. So we try to avoid using different methods, like UV sterilization we do. And then we also do ethylene oxide sterilization, where it's mostly done in hospitals. They do ethylene because it's a dry method. It's not a wet method, so it's easy to do. So we try to do different sterilizing methods to keep them not contaminated, that all. So we do that all. So no more questions on bone? So shall we move on to cartilage, or it's too boring that we will skip the cartilage? Because I see it's 1 o'clock and then I think you guys are hungry, right? Yes or no? I hear some yes and then some no. So yes or no? Hungry? So we leave the cartilage part? Yes or no? So we will leave the cartilage or we will talk about the cartilage? So I'll quickly go through what we have done in cartilage. I'm not going deep into cartilage about it, but then we will see what I have done in quickly. So this is a basic why we wanted to work on cartilage. So most of the people have, this hip cartilage, osteoarthritis diseases, and then it's 350 million has been affected, and it's most of them are less than 40. So it's 20% 72% is less than 40, but what we have in a cartilage, treatment modality from 1975 to so far, currently we are working on 3D bioprinting techniques, but then we started with something allograft. So what is allograft? What is autograft, allograft and xenograft? What is first what is autograft? I could hear cell, but yes, so it's from your own human body, self. So but this is your autograft. And what is allograft? Yes, so it's from human to human is your allograft, right? And what is xenograft? Same thing. So what we had previously is your allograft, and then we moved from autograft to C, but then what happens in autograft is most of the time the person will not have a perfect match. Either the person have a already disease history which we cannot isolate either their tissue, so that is we lack in most of the time in autograft. But allograft, what happens is that there is an inflammation or infection happens. Let's say you take from some other people, the immune reaction is so faster, most of the time it will get rejected in case of autograft. And then we neglected all of this, then we thought of, okay, let's go to tissue engineering to see what happens. So what they usually do is do a microfracture if there is osteoarthritis happen, because there is no effective cells, active cells in it. They want them to be, start coming to the site and repair, because it's cartilage, there is no blood circulation. So do you know that your cartilage doesn't have a vasculature? Do you know or not? So your cartilage do not have vessels in it. So there is no active cells going inside to your cartilage. So repairing a cartilage is very difficult, because there is nothing going, because there is no vascular, so that it will not penetrate from, from other organ to your cartilage. So it's complicated. So we cannot have any cartilage specific cells or cartilage, precursor cells to go there, which is complicated. So what they usually do is, they microfracture. Basically it's like a needle. They fracture in the sense they poke your cartilage so that it is the cells which are inside from your bone marrow and bone surface will start coming out and then be in contact and start repairing on its own. That is what they do in microfracture, but most of the time it's not effective. If they are very elderly people, let's say 50, 60 years, they are not going to have active cells in it, so it will not happen. So then they talked about 3D bioprinting and other methods of using tissue engineering approaches to work on cartilage. So the major problem we have is uncontrolled degradation. Let's say we put some material, I talked about already that the degradation happens in an uncontrolled manner. I want them to keep for 1 month, but then what if they go degrade completely in 1 week? So that is what happens most of the time. So this is uncontrolled degradation, and then we are not able to monitor them. So whatever these clinical studies or surgeries happen, they put, and then we think that it will repair, right? That is what we do. We will implant it, and then over 1-2 months we will go to the surgeon and see. In case of metals, it's easy to do your MRI or CT to see what happens between your bone, but in case of cartilage it's too complicated because it's between your bone, right? So it's very difficult to see what happens in cartilage over time. So that's what we wanted to see, how we can do monitoring. And then also there is a mechanical mismatch between your cartilage. Even though cartilage is soft and flexible, they also require some stiffness and then strength to support your load condition. So that is also happens. There is a mechanical mismatch between what we make in lab and what needs for the clinical. And then there is poor integration in the sense of scaffold and cells between their interaction is too low, because we make something in bio inert and which is not going to have easy communication between them, and then the growth, and then the differentiation of cells. Sometimes the cells, so cartilage cells, it's called chondrocytes, which are very difficult to maintain in a laboratory condition. So if let's say these cells takes at least 28 days to differentiate from your mesenchymal stem cells to cartilage, but then after 28 days there is a higher chances they will go back, they will de-differentiate also. So that is also another clinical challenge we work on. So how we can able to do this? So this project was done with St. Vincent's Hospital in Australia. So the advantage is that it's a hospital lab. So what we do is a clinical problem, and then we try to address by engineering. So whatever we get a suggestion or everything from a surgeon. So we talk to surgeon, we see what is the problem, and we try to address. So that is where we come to this project. How we have a scaffold, we see their degradation over time, and we also wanted to see them easily, like a non-invasive monitoring, because lot of time what they do is they put in, after some time they will ask you to come back and then they take a biopsy, right? So what they do with the biopsy? What is the biopsy for? Yeah, so they try to see the infection, they try to see the specific type of cell needs to be there. Is it present there? Is it some immune cells come in, or some cells which needs to be there? Your stem cells plays in role. So that's what we wanted to do in biopsy. But then your cartilage is so small and thin, your affecting with the biopsy again is going to regenerate faster. So it's going to take some more time after a biopsy also. So what if we have something non-invasive method which will, easily be monitored after the implantation? That is one of the major criteria also we talked about here. So we have something, fluorescent scaffold. We talk which is green is a flash, which is a fluorescent scaffold. So we made a fluorescent scaffold, and then collagen too, as I mentioned already, each organ has a different collagen. So cartilage has collagen too. So when there is a degradation, so you see on the other corner there is a black space available. So that means the scaffold is degraded, because it's a green fluorescent scaffold. When there is a green, you don't see a green fluorescence, that means there is a secretion of your collagen happens. So the cells inside them start degrading the material, and then they will, secrete these collagen, extracellular matrix, and then they accumulate there. So that is what we wanted to see over time. So this could be correlated your in vivo, right? We see in vitro, and then we try to correlate them with your in vivo. So that's what we have done here. We put some stem cells, and then we try to see their scaffold degradation with differentiation in terms of chondrogenic progenitor cell, which is called chondrocytes, or differentiation of cartilage specific. So we try to address two different problems. One is the improving the mechanical strength of the existing scaffold, and then the other one is having two dual tracking system. So we have one material which is already, labeled with green fluorescent, but then what if we have two different material? So that's what I worked on it. So having a two different material, so the one at the outer I call it cage, or it's a honeycomb structure, which is a typical hexagon structure, okay? And then we have some hydrogel in it, which is also having some fluorescent molecule. So there is a two different fluorescent molecule, which is FITC is green, that's why I put it in green. And then there is rhodamine, which is red fluorescent molecule, and then we have two of them, and then we see their degradation and ECM over time. So I'm just not going deep into it, I'm just going to run over. So we try to able to make these synthetic polymers, which is PCl is polycaprolactone, and then PLGA is polyglycolic acid. We try to attach them with a photocross-linkable monomer. So you buy a commercially monomer, you attach them with your fluorescent molecule and see are they able to, attach to that polymer and able to release over time. So we did a couple of studies to see it happens, and then try to fabricate some 3D printed scaffold. So we try to 3D print this hexagon type of structure, and to see, so you see this, there is a color change in visibly, and then with, so you guys know gel dog? Same thing, we use the gel dog. So we put it under the gel dog and then use the FITC green fluorescence, and then see there is a green color. That means it is green fluorescent polymer or material, you made it already. So that's what we did. And then we try to correlate, as I mentioned, the degradation with fluorescent release. So when there is a degradation happens, there is a fluorescent release happens. So you measure the fluorescent release using plate reader. Do you guys know plate reader? Have used plate reader for absorbance fluorescence, right? Similar thing. We use the plate reader to measure their fluorescence, and that we correlated with their mass loss. So when there is a fluorescence release, there is a reduction in their weight. That is what we correlated. And over the time, we did a linear regression to see how much is correlating, and we see a perfect correlation between the release and the degradation happening. And then did a couple, just a preliminary study to see that this synthetic polymer is actually, biocompatible with cells. That's what we did. So what we have now is the outer structure, which is my hexagon, right? I have that. So what I want is the internal structure, which is my hydrogel with cells. That's what I wanted to make. So the second study what I made is having the first one where we talked about categorizing all of this on a category on topmost, and then the second one we have it on figure B is about talking about making this middle part of it, which is a hydrogel scaffold. So we made red fluorescent hydrogel scaffold, which is on gelma rhodamine. We were able to make them, and again this is also photo cross-linkable, so when you shine light they will cross-link faster. So advantage of photo cross-linkable is like 30 seconds, right? The nail art is like how much time they use it for cross-linking? What do they do in nail art? They cross-link your photo resin, right? So it's quite faster, it's like seconds it happens. The similar thing happens in here. It's like 30 seconds the whole scaffold will get cross-linked. So this is the same thing. And then in case of the C, the last one, what we did is we incorporated this cage and then your cylindrical hydrogel, we put them together and then we made this composite or reinforced scaffold. So the outer layer is mechanically stable and the inside part is hydrogel, which is soft and squishy. So that where your cells can present, and then the outer part is where your mechanical support comes in. So this scaffold is basically to provide mechanical support and then also some regeneration capacity from the hydrogel itself. So I did a couple of study. I'm not going to run this. This is just to see how this PCL, like the fluorescence release is categorized with their mass loss and all. And then we also tried to talk about different scaffold fabrication because this is a 3D printing technique. We wanted to see the feasibility of the technique on these fluorescence scaffold. So we performed a lot of different structures with that. One is my typical cage structure, and the B is the log pile structure, is nothing like printing a square with some inside structure with same square, and trabecular is basically a trabecular bone. So as I mentioned, it is a hospital, we have a collaboration, we get some human sample, trabecular structure sample, and we try to replicate it. So we try to print those trabecular also and see how the degradation happens. So we categorized for all of the structure and correlating that, it's like correlating that your mass loss and fluorescent release is similar in both the cases. So now we have that categorized. Then we have gelma, which is your hydrogel part. The middle part is being categorized. So then what we have is the whole structure. So the whole structure, so the degradation happens under what condition in body? How the degradation happens? How the degradation happens? So with the help of enzymes, right? Same thing. What I have is called collagenase. The last ASE suggests it's enzyme, right? Collagenase. So that's what we did. So we used collagenase so that we are mimicking an in vivo condition in these scaffolds. So when you treat these scaffolds under collagenase, they will have a similar mimetic environment what happens in the body, and then that is similar to the degradation happens in the body. So we tried to do their degradation under collagenase and then found out that over like 21 days there is a degradation happens in case of the gel, which is your red color part, because it's hydrogel, right? Hydrogel degrades faster. But then what I have, the other one is a cage structure, which is a synthetic material. It doesn't degrade faster. So it's suggesting that the hydrogel will provide regeneration capacity, but your cage will provide mechanical support. That is what we were also able to see under degradation, that over 21 days it's still stable. It doesn't degrade faster. So that is what we found here. And then we also found their mechanical strength. So it just suggests that this reinforced one at the last with yellow color is higher mechanical strength and suggesting a better scaffold. So we did a couple of same studies to see how the fluorescence is correlating it. So if you see here is, as I say, it's a chemi-dog, is gel dog images. So you see a gelma, which is a red fluorescent. When you see this reinforced or composite scaffold, we were able to see two different scaffolds in a single image. So that is our idea. Two of the fluorescence molecule, we wanted to release them, and then we also wanted to see their release over the time to correlate their degradation. So we were able to do this all under with cells. So these all are the images with the cells. So these all are again mesenchymal stem cells under some differentiation condition, and over that time we were able to see their differentiation happens, and we were also able to do some scanning images to see it's still there, and then the degradation happens over the time and we correlate it. And this is one of the fancy image we have it. So we recently had some light sheet imaging microscope. What happens is that you provide a complete 3D scaffold in it, stained, and then you able to get all these 3D images. So whatever we mostly get is like 2 to 2.5D. We don't see a complete whole scaffold in the structure. But what we have is basically this 3D images. So the whole 3D scaffold is imaged under that. So we have this red color is basically your scaffold, but we have this green color. You see, these green color are collagen 2. So cartilage has collagen 2 expression. So this collagen 2 is being expressed. So we were able to see in this image also there is a collagen 2 expression throughout the scaffold. It's not only a surface or something, it's completely present. So when you see on this image, it's quite difficult to see, but then where there is a black background, wherever there's a black background, you see this green color happen. So it's suggesting that the cells are degrading the material and then they start accumulating or secreting their own extracellular matrix. So that's what we found in this scaffold, that they were able to support the differentiation of cells. During this differentiation process, they were able to produce the secretion molecules for supporting their regeneration. So that we found in these scaffolds. So the overall summary in bone is we were able to make a bone mimetic scaffold with significant mechanical strength for bone tissue regeneration. And in case of cartilage, we were able to make these reinforced structures, and then we correlate their fluorescence release with their degradation and then see their differentiation of cells in cartilage repair. And then we found that in case of cartilage repair, and also it is a high throughput technique we call it, because just with the fluorescence release using a plate reader or just using your gel dog or chemitop, we were able to see their structure. The whole structure will be visible. So let's see that you were able to provide the similar wavelength of that into your cartilage, and then we were able to image them or monitor them. So which is a way easier than your biopsy, right? So that's what we wanted to have. So in that case, we will be able to have a non-invasive monitoring system to see how these scaffolds work under in vivo or clinical conditions. And these are the groups which I worked, and of course I would like to acknowledge all my research institution, research lab, and all the funding support, and thank you all. I would like to take any question. No questions? Full sleepy and lunchtime? I'm like, ma'am, please leave me. I want to go for lunch, huh? Huh? So should I ask questions then from your presentation? Because you guys are not asking me questions, then I should ask you questions, right? Now it's like everybody woke up after the whole 1 hour of talk. Now it's like, okay, active, let's go for lunch, right? So if you guys don't have any question, but then if you have something, I have my email ID, write to me. There is a web page, just find out, and these papers are already there in my web page, and then you can try to reach out to me, okay?
I just wanted to know any successful translational attempts.
So in case of translation, everything we do is under animal testing, and some of them went to like clinical, which is also in TRL 5 or 6, haven't not completely completed FDA approval, and when we have not went to something related to clinical yet.
And also another thing is about IPSCs. Can IPSCs be the source for scaffolds?
We can also use IPSCs. The only thing we are not using IPSCs that India is not very established IPSC cultures have. Not many researchers have IPSC facility and IPSC, the cells, because it's very cost. It's too much to handle the IPCs. Like mesenchymal stem cells is easy, but then handling IPCs is like too much of like labor thing, and then also too much of consumables to buy for us. So we are not completely done to IPSC so far. There are few labs has IPSC, but yes, possible. Sorry, sir. So what we try to match is basically we understand what is the thickness of the samples inside the vascular, and then we try to match the same thickness of the layer. Like let's say the vascular width I wanted to make is 3 mm of diameter with the wall thickness of 1 to 2 mm. We try to implement them on a similar way to have them the wall thicknesses too. So the overall diameter is like 4 to 5 and something. Yeah, that is possible. But the different techniques we might use it. It's not the similar 3D printing, but there is different type of 3D printing we use it to make that. That is possible. Yes. So these micro nanoplastics in regeneration are in disease. That's the question. So I don't see anything in regeneration, and I'm really not sure about these plastics in terms of regeneration, which is really not my research field yet, but I do see there are people work on lot of this nano and microplastics and repair, not even repair, mostly I do see about in disease. How these plastics, they go into your blood room and being accumulated in all the organs. That's what we see, but it does affect your normal functioning, but I'm not sure about its regeneration in plastics, which I'm not sure about it really. So this is a very growing thing. That's good. So the institute I worked in Los Angeles is also, they have a this called O-meat. They had this 3D printing of meat. So they do also make meat printable or synthetic culture meats. There are a lot of companies and work on it, but there are very few come into market, I remember, but nothing in India so far. There is few companies in US, there is one in Australia, I know, but it's not very I see it's not people not accepting it so much, but they do, yes. Yes, it is possible. They do work on it, it is possible, but then we don't know the long-term effect of, because it's all happening in your culture, right? And we wanted to take it into your body as your food source. So we do know at certain level, we do have this all the regulatory, we follow all these regulations to do it, but then on the long term, how it is going to affect is not, we have not known yet. That is the still, ongoing research, still people also work on it. That is also one part of it, people's to work, but then it's possible to do with 3D printing. So that's what the previous, I'm not sure is O-meat is still working on it, but the company is called O-meat. They were working on these printable synthetic meats, yes.
Students, this 3D bioprinting is a very current concept and is a fascinating dream for all the scientists, and for patients it is going to be a big boon. So it's a splendid opportunity that you all have got today to listen to the expert lecture. So thank you, Dr. Meenakshi Kamraj.
Thank you so much.
I request Dr. Panchami, Assistant Professor from Department of Chemistry, Biochemistry, to present appreciation letter on the behalf of MSRCAC and KSTA. Thank you all, and you can proceed with lunch, and those who have registered for the quiz, you have to retain. Please wait for the announcement.
Students, those who are registered for the quiz competition, please stay here, and rest of the students can go for lunch. Only the students who have registered yesterday can stay here, and rest of the students can go for lunch.
Be quick. Be quick. Except the registered participants for the quiz, rest of you please go for lunch. Only those who have registered yesterday. Only those students have to be here.
24. All the registered students, please keep your mobile phones back at the back of the auditorium. Please keep your don't keep your mobile phone, keep it inside your bag and keep it at the end of the auditorium. The back side. Back side of the auditorium.
Backside chairs, you can keep your bags, everything. Except your pen, nothing should be there.
The first team, Divyashri, Bindu.
Who's Divyashri and Bindu? You can sit, come and sit here.
Nandini Soumya. Nandini Depsinga Soumya. Dimple and Maitri. Single name? Is it a single name?
Sushmita and
Rayanka, it is.
Rayanka Gari Nishchita from Reva.
Who is that? It is your name, Rayanka Gari Nishchita.
Nishchita.
Revati Pratibha MSR. Sahana Bhumika MSR. You start from here.
Only
Bring
two
your
should
pen.
sit here.
Then Indrani Lavanya from Reva. Aman Vishaka Kristu. Nitin Akshay from Kristu. Arya Gopika MSR. Deepthi Chaya Reva. Nakshatra Nancy from Kristu. Harshini Shubha from Kristu. Pooja Jyotika from Reva. Amulya Swati from MSR. Mayura Ashrita from MSR. Then Hemapriya Gomati from Kristu. Swati Vaishnavi from MSR. Sneha Priya Sahana from JSS Mysuru. Revati Kirtana from MSR. Sumeda Aditi from MSR. Hasta Rakshita MSR. Then Ankita Kushi MSR. These are the teams. Total there are 24 teams. Sahana Bandilwala. Sahana Spandana. MSR and Reva. Okay. ಎಲ್ಲಿ ಬರ್ಸಿದ್ರಿ ಯಾವತ್ತು ನೋ ಬೇಡ ಇಲ್ಲ ಅಲೋ ಮಾಡಲ್ಲ ಯು ಶುಡ್ ಹ್ಯಾವ್ ರಿಜಿಸ್ಟರ್ಡ್ ಎಸ್ಟರ್ಡೇ ಐ ಆಮ್ ಸಾರಿ ಫಾರ್ ದ್ಯಾಟ್ ವೈ ಫೈವ್ ಆಫ್ ಯು ಫೈವ್ ಅಲ್ಲ 6 ಟೂ ಇನ್ ಅ ರೋ ಟೂ ಟೀಮ್ಸ್ ಇನ್ ಅ ರೋ
You can shift to other this thing, the middle two. You can shift that side or somewhere. You people can shift to the last.
ಅವ್ರೇನು ಕಾಪಿ ಮಾಡಲ್ಲ ಬಿಡಿ ತೋರಿಸಲ್ಲ ಬಿಕಾಸ್ ದೇ ವಾಂಟ್ ಟು ವಿನ್ ದ ಪ್ರೈಸ್ ತೋರಿಸಲ್ಲ
Don't keep your mobile phones. Keep it outside.
ವಿ ಕೆನ್ ನಾಟ್ ವಿ ವೋಂಟ್ ಜಸ್ಟಿಫೈ ಮಾಡಲ್ಲ ಕಾಲ್ ಹಾಂ ಕಾಲ್ ಹಸ್ತಾ ಅಲ್ಲಲ್ಲ we want some volunteers to monitor.
Kachin karsana nanka. Students, if we see any cell phone, even by chance, you will be disqualified. Okay? No material should be there, or else it lead to disqualification.
ಆದಿತ್ಯ ಅವರು ನಿನ್ನೆ ರಿಜಿಸ್ಟರ್ಗೆ ಹೇಳಿದ್ರಾ live done. No. We want a few volunteers from outside to monitor. Okay. Question papers.
Question paper first is the rules and regulations. So these are the rules and regulation for the today's quiz competition. So the quiz competition is based on the first 5 lectures. 2 members in a team is must. Quiz is MCQ based, written test for 40 questions. Duration is half an hour. The winner will be assessed based on the question answered from all the lectures. The top 3 winners will be awarded cash prize along with a certificate. All the best.
Now the time is, what's the time now? Now the time is 1:33. You have to return your answers by 2:03. You have to return the papers. Yeah, one per team.
One per team, one paper per team.
Please write your name, both of your name, department and the college.
If you submit without name, will not be able to assist. Please write your name at the top of the paper. Name, class, and then college. afternoon, everyone. Welcome back to the next session of the guest lecture today. We are privileged to have with us Professor Dasappa sir, professor from National Center. Professor Dasappa sir, Interdisciplinary Center for Energy Research, IISc Bangalore. I would like to request Dr. Pratik sir to kindly introduce Dasappa sir to the audience.
Thank you. Gives me immense pleasure to introduce Professor S. Dasappa sir from Interdisciplinary Center for Energy Research, IISc Bangalore. Professor S. Dasappa sir currently in advisory role at Interdisciplinary Center for Energy Research and faculty at Center for Sustainable Technologies in Indian Institute of Science, Bangalore. His major research area is in combustion of different forms of fuels like solid, liquid and gaseous fuels with the emphasis on energy conversion process. The R&D activities address both scientific and technological challenges. The research output have contributed to the engineering domain by addressing the process to product philosophy, including sustainability through translation research. In recent time, Professor Dasappa sir has been involved in R&D related to the biomass and high ash coal gasification for the power as well as chemicals. With over 220 publications in various national and international journals, conferences and edited books and 30 patents, he has been involved in technology transfers across the globe. The global presence through his partnership and technology transfer and policy influence be a testimony to the importance of relevance of R&D as well as technology development efforts. The current research in the area of hydrogen from biomass for PEM fuel cell application is a unique and novel process and is expected to make big impact on production of green hydrogen. Professor Dasappa sir has a joint project with Indian Oil Corporation to set up the first green hydrogen from biomass for PEM fuel cell bus applications. With that, I would like to request Professor Dasappa sir to take over the session. Thank you.
I request Dr. Lakshmikant sir, assistant professor, to give the sapling to Dasappa sir.
Thank you for the introduction and happy to be here to share broadly the kind of work what we have been doing. And before that, I thought I was given the topic renewable, green and sustainable. I think these are keywords which I thought I should share with the friends, young friends sitting here. And you have already gone through the sustainable development goals and a range of things which are sort of relevant in the current context of what we have been going through on various things. including the climate change, what I think could be one of the subjects in your core area of maybe activities what you have been doing. And for most of us, when we really look at renewable, green, and the green, the terminology green, I was surprised to see this title given to me, but I said, okay, let me use the title without modifying that one and try to understand and try to interpret for you. Renewable, I think we knew broadly when we call renewable the way it can happen. Maybe it could be an interaction also, it could be a dialogue between you and me. And green energy is something which has come in the recent time. It's not that we have been really aware of this term called green energy, which was there from earlier pathways, but I think something which we have been really looking at in the recent time. And the major thing which is about sustainable and related aspects, over the last decade, I think it has become a prefix for everything. If you really look at any, earlier it used to be nano something, nano, for everything you had a nano coming in, and the other jargon which is going along with this prefix, sustainable transport, sustainable energy, sustainable something, engineering. These are various things which is happening that I think it's also important to understand what is the distinction amongst these one, renewable, green and sustainability. I think broadly that also becomes very, very important when you really want to look at how various options can be really looked at, why people have been talking about all these things. Now along with this, I will bring in another yes which is important, which I have not marked here, is security. We will also discuss why that becomes very, very important. I will try to concentrate more on energy, giving you a glimpse about what you have been really reading, seeing, and also looking at the public information which is available and the way the country is progressing across the globe. It's nothing to do with our own country, maybe across the globe broadly on energy scenario. Why all these keywords become very, very important for all of us to consider and move forward. Yeah, sorry. Maybe I thought I should also introduce the institution I come from. Most of you would have heard about this one, but I thought it would be nice to get to know. This is from Indian Institute of Science. Maybe you know Jamshedji Tata, and there's the Government of India and also the Mysore royal family. I think this today, if you really look at today management jargon use, it was already a public-private partnership which took place about 100 years back. That means you have private party as Jamshedji coming in, Government of India as a government, and the royal family coming as somebody who looks at really looking at what needs to be done. I think based on the land which was made available from the royal family, Jamshedji thought that Bangalore could be the place for the hosting of an institution. And the charter, it was basically looked at, conceived based on the establishment of John Hopkins and Caltech. Even today it's slightly a unique thing compared to most of the institutions, higher educational institutions in the country. This IISc is very, very different in its approach. That's something which I thought I should do. The earlier department were basically chemistry and electrochemistry, and most of it was designed to meet the Indian requirement. If you really look at the so-called the founder's mandate at that point of time, was institute is designed to promote original investigation in all branches of learning and to utilize them for the benefit of India. I think broadly, if you really look at, this is how the entire institute, if you people would have known this soap factory, Mysore sandal soap factory, it originated from here. The other extreme which you know is related to the so-called the Wipro. It also originated from this thing. And quite a bit of activities which have taken place has made IISc to be the mother of all institutions, several institutions across the country and probably many parts of the world. I think that's something which I thought in today's context probably one of the foremost sought academic institution in India, and probably it's something which promotes both education and innovation. That's something which probably I think most of you would be really looking at these things. And IISc for some reason had never got into undergraduate until very recently. Over the last maybe about 5-6 years, there is an undergrad program going on in about basic sciences which is there. Otherwise, basically it used to be a postgraduate and a PhD program. We had totally about 4,000 students and about 500 faculty member. Broadly, this is how the distribution is all about, and there are about 42 departments and centers, and these are the various divisions which really look at, and I should say that all branches of science and engineering are available on the campus. That's something. In the recent time, I think medical sciences has been brought in, wherein it's going to be integrated with this science, engineering and medicine together. I think that's a comprehensive way in terms of the way things are being looked at to see how we will be able to do that. And there is going to be a hospital adjacent within the campus to make sure that all the necessary other research activity can take place. This is just to give you a glimpse about what the institute is all about and where we stand with respect to institute. And in terms of, I should say that probably it is one of the most sought institution and probably the top institution in India, and as I told you, it also has become mother of several other institutions kind of situation. Let me just come to, have you heard of this term renewable? What does it signify? Because we have been using this word renewable in various contexts actually, especially when it comes to energy. Probably you would have come across certain things with respect to energy. Where do you place renewable as? Is fossil fuel, petrol, diesel a renewable? Why? It will exhaust. Okay, maybe we'll just go a little deep to understand, and then if I were to ask you what is sustainable, is there any difference between renewable and sustainable? What is the difference? It may look very trivial, but I think this terminology gets, I should say, as an interplay depending upon the situation. People really look at this one. I think if you really understand the boundaries under which these terms can be utilized, it probably will also help you to see how you will be able to conceive certain things in a given situation. When we are pushed to, when did renewable come into picture? Maybe over the last 3-4 decades when people have been, otherwise we have been using renewable. Most of our, if you really look at the way we dry our clothes through sun, it is some form of renewable energy coming in. The way you dry if you go to rural side, the way they dry the grains, you do the chaffing and you put the grains for drying, whether it could be on road or a pavement, still it is used. I mean, somewhere or the other, as a civilization, we have been using all right sources, but because of the advent of the fossil fuel, some of these things have lost their impact, and now it is gaining importance over the last maybe about 5 decades or 4 decades kind of situation. And I think somebody mentioned, yes, it has its own limitations. Maybe I'll just go through a little more definition so that it becomes clear for us how do we move forward and whether some of these things make sense. I think we can debate whether this is right or wrong. The meaning is the energy source that naturally on human time scale. That means it just over a period of time, it's much larger time scales compared to, now you should really look at the relative time scales. For example, how did petrochemical or fuel come into existence? It was a source for all our, let us say, diesel, petrol or gasoline and various other things, maybe natural gas. Fossil fuel, it is some dead of plant animal matter over thousands of years. You try to do that and you are exploring, and the time scales at which we are using that compared to the time scales at which the growth of those things have taken place is not in a relative manner. You can call it as renewable. Sustainability will come separately. I thought this would be very useful. Will the resource run? No, not run out. later on. I thought I should just give you some of these glimpses. What does it not guarantee? Low the emissions, low ecological balances, social acceptability. Broadly speaking, in the context of what we have been going through now in the present scenario, when we are really looking at energy security as a point, now you must have seen in time, everybody is fighting for energy now. Globally, everybody wants to see and secure. I don't know how many of you follow what happens internationally. If you really look at about at least 3 years back when Ukraine and Russian war took place, people in Europe suffered a lot because they did get the gas, and for them being a cold country around this time, it is very difficult to survive. For them, gas becomes primary important for heating. The electricity may not be very important. For them to keep the place warm becomes very, very critical. I think that's where the people started that can I become independent? Can I have security? That's the reason why everybody is trying to see. Even currently, when yesterday or day yesterday, whatever we have done some signing, people in the US. At the end of the day, the government of India is saying energy security, meeting the needs of the energy becomes very, very critical. This is there, irrespective of whatever happens to the climate, while that could be a goal, but security becomes very, very important because energy becomes primary commodity for all developmental activity. At one end of the spectrum, we are talking about doing AI/ML. For AI/ML to run the way it has to run, the gigabytes has to run, you need energy. I think because everything becomes, during that process of generating energy, can it be green? Can it be low emission? These are the questions which are being asked, and what are the various technologies which will satisfy that. To that extent, some of these things would help you to analyze, maybe also assess what could be done, where it could land up kind of situation. I think that's something which we need to do. And these examples which you know, whenever we talk about renewable, you know it's one thing which becomes very, very clear. People say solar, next comes wind, next comes hydro. Hydro under certain boundary conditions, if you are looking at the multi-megawatt, it is not so-called environmentally friendly, it will not fall into that one. And there are another component called, some of you who come from biotechnology, it's not a new word for you, biomass, and other things with respect to the so-called tidal and geothermal. These are all very, very little explored in our country, but these do make a part of the broader contours on which the renewable energy portfolio is being looked at. In India, I think we are strongly concentrating, and these two are the primary important, and depending upon where you are using this, depending upon the streams, hydropower, it starts with, maybe I have a few slides to talk about that one. It is the technology wherein you use water motive power as something to drive the turbines and generate electricity. Similarly, when we want to really look at green energy, I said that the definition also becomes very, very important. How do you define something? I think keeping that in mind, maybe I'll just go through something similar. Energy that causes minimum environment. Previously, we said it may not have any major impact on the environment. I will come to some numbers as we move forward, but and especially low greenhouse gas emissions. That's how it is getting defined. If you are following what is happening in Government of India today, everywhere there is an hydrogen, the word being used. And whenever hydrogen is being used, we also use in our countries a lot of hydrogen. But today we are prefixing with something called as green hydrogen. And what is that green? To some extent, this definition will satisfy that. Can it be very, very, I hope you understand what is GHG emission. This is something which we need to make sure as greenhouse gas emissions, which leads to what we call it as the climate change, impact on climate and various other things. We can discuss that as we move forward. But again, most of these things what we mentioned comes under this one, but we need to make sure that other things do qualify. I'll just touch upon that part as we move forward. And what might not be green is when we use a residue in such a way that we are depleting at the rate at which your much more than at the rate at which you are trying to utilize. For example, I have a forest wherein I cut the forest for energy purpose, but I don't grow the forest at the same rate. Then you are trying to deal with. I'll come back to you because some of you come from biotech. I can also give you a scientific reason why this becomes a very, very important. And maybe large hydro, which I mentioned, it has a major impact on the settlements and people moving around. It's not going to be green generally, and it cannot be sustainable too. That part we will also look at. And maybe solar, yes, I'll come back to recycling. I have one to talk about that one. I'll give you some information about that one. Finally, the term sustainable, I think, is also part of the sustainable goals which has been listed as a part of SDGs, and it also becomes important how do we really look at this part. If you go back in the same terminologies, viable long term across 3 dimensions, that means here we are not looking at energy per se as energy. We are looking at can it impact the environment, can it impact economics. There are 3 or 4 pillars each one people talk about. Societal relevance also becomes very, very important. I think when you really look at under these dimensions, the topic covers most of it which needs to be really looked at, and if you see, it can impact most of the goals what has been set out here to see how energy can contribute to each of those sub-activities which has been listed there. Again, can this system last for decades without breaking the nature? I mean, these are core ideas and something which probably you can carry forward, think and trying to see what could be the way we will be able to address. Because everything what people are projecting could be sustainable, need not be sustainable. For example, our earlier pathways of everything used to be sustainable. For example, if you really look at one simple thing is, most of the time people used to use a handkerchief made of cloth as a part of your carry-on wherever you go. Today it has got switched into the so-called a paper napkin. Which do you think is sustainable? I mean, these are all small things, but it can make a big impact in terms of the way you understand, the way you can interpret this one. I mean, that way there could be many things what we have been doing in nature. And if you go back to certain activities what as our predecessors were doing, most of it was sustainable, most of it. But with the advent of technologies or adopting something which has come from overseas, we are trying to create an imbalance. Maybe as society grows, things will happen, but is there a balance which you can establish between these two so that at the end of the day you can create an environment which is there? Today people have been talking about everything about weather is changing, so-called the summers and the winters are not distinctly clear what is going to happen. At the end of the day with GHE, people are worried about the melting which can happen in the Himalayas or sinking of islanded land. I mean, various things which are happening. I think these impacts have really made sure that whether people will be able to look at these things and trying to provide solutions. That's the reason why there are large bodies which have been really looking at these. Whether at one end of the spectrum, I hope many of you who do biotech, they say that growing paddy is worse for climate. Do you know the reason? It provides more methane because of the microbes acting in and around the vicinity of the growth pattern, and various things people have been talking about, and we should now not look at that sustainability as far as India is concerned, security as far as India is concerned. That's how we should start looking into. And if you go back today, even in our recent budget speech, nuclear is gaining importance. Is just again because of security. If you can have a module which can generate, there are safety issues and various other things which will come in, which will dictate the sustainability of those operations. But if you take care of many of those things, is it going to be sustainable? People have been able to argue saying that it could be sustainable as long as safety is going to be taken. I'm just trying to give you examples in such a way that you will be able to measure up to see that if given an opportunity, how do you distinguish between these 3 very subtly different from each of those definitions. I thought it would be nice for us to really look at, and there are issues when you want to really look at this. That's the reason why this has become very, very important, wherein all the countries are driving this not for anything else but to make sure that for you to become sustainable, there is something which you need to compromise with. It could be financials, it could be something else. If you want to do that, if you want to make sure that something is clean, you need to put in that additional effort. One simple example is, today we are putting all our garbage into landfills. I think this problem must be evident across the country. It's nothing to do with Bangalore or Karnataka, everywhere. We don't have a solution. Can there be a sustained solution? Today there is another terminology which people have been talking about called circularity. I hope you have heard of this now. I mean, some of these things become very, very critical when you finish your degree and move on to another space wherein your knowledge base with respect to your subject is very, very critical. But surrounding that, any of these things can make a big impact in terms of how you can present your case to a given situation and trying to understand and make sure that that happens. And it's very, very clear, if there are going to be, for example, solar, there is one very, very critical thing which people talk about. In wind, I'll talk to you something else. If you are going to put solar everywhere, there is no land for farmers, then it's not a right way to look at. It's not sustainable. These are simple examples, but I thought it would be nice for us to be clear. And when you put all these things to a framework and do a comparison, in a time frame, renewable resource availability focus would be emission and ecology and long-term health checkup. That means system health. That means, can you really do everything in such a way that it doesn't disturb the entire ecosystem? Time frame, in the time frames what we look at, this is going to be much larger time scales, includes economics. Yes, this can carry take, that's what I said, pillars of sustainability. If you look at any book on management, once they say sustainability, there are at least minimum, they say 3 verticals or 4 verticals which will dictate the entire thing. And policy buzzword, medium, high risk in terms of risk, extremely low, because you need to really satisfy many of these things. It's not going to be easy. People may be talking about, and especially a country like India with so much of diversity, making sure that you really take care of these things under the banner of sustainability is not going to be easy, but I think there are progress done in various aspects. Now coming to the country and trying to see whether I introduced another word called security. Let us just see that we know that most of our power, more than 50% of our electricity is coming from coal. Basically that's what is the thing. And along with that, there is some percentage of hydro coming in, extremely small percentage of renewables, and nuclear is very, very trivial. I think broadly this is the kind of distribution what we have. When we want to generate electricity, we have coal as the thing. We consume roughly about 900 million tons of coal. That means this is the kind of coal. You take coal, generate steam and generate electricity. Broadly that is the kind of process. And we emit, whenever you burn coal, C plus O2 will become CO2. That is the emission which people are talking now. That's the reason why they say you should reduce the amount of coal, because CO2 is increasing. I gave you an example. The rate at which we are putting CO2 is not getting fixed. What is happening is gradually the rate is increasing. Because of CO2 rate increasing, it is still in ppm. Maybe if you go through any of the so-called climate change reports, still in ppm, but that has made a big impact in terms of the overall climate across the globe. I think that's something which we need to be aware of. And every kg puts in some amount of CO2 which people are looking at, can it be avoided? Basically that's the thumb rule. And in terms of decarbonizing the sector, India is seriously contemplating on solar and wind as the so-called opportunities which is available. Because being a tropical country, we are blessed with sun for major part of the year. That should not be a problem for us to really get that going. Everything is fine. Are there going to be problem? In this state only we generate sometimes more than 50% of our electricity from renewables. Both wind and solar provide more than 50% for many months in our country, in our state actually. That's something. Similarly, there are a few other states wherein, except that there are some limitations which do come in when we really look at broadly the sustainability aspects from any of these renewables, but still they pave way for the so-called mitigating the issues with respect to electricity. For example, Germany has done quite a bit with respect to solar, but they had problem in terms of balancing power. Whenever there is solar, you pump into the grid, but during night you don't have electricity, and that's the time wherein people have been talking about today, can we store? Batteries are becoming critical, the research on batteries are going up, and there's quite a bit of work being done on batteries. I think this is how each is getting linked to another technological development to make sure at the end of the day, is it going to be sustainable is a question everybody is asking. Each time we are going to put a component, and that component beyond the life, what will happen to that one? That's where the sustainability will become very, very important for us to address. Second important thing, we all use all vehicles and everything is nice, but we still import close to about 300 million tons of oil, about 240 million tons of oil and about 60 million tons of gas. I think close to 300 million tons is being imported, something as crude, something we may also export as finished product, but this is the kind of consumption. And most of the time this is for the liquid fuel is for transportation, heat and various other things. Gas is also partly for energy purposes, and finally it goes into refining and other things. Basically, I'll give you also example where this is getting used. And if you really look at these two spectrum, this is where most of the energy is going in our country. What we really looked at, everything is a fossil fuel, everything is a very conventional. There is also, prior to renewable, people were not looking at non-conventional terminology. Conventional when it gets assigned to all fossil fuel technologies, non-conventional other than that, but with time I think that non-conventional has become the ministry, which was Department of Non-Conventional, has become Ministry of New and Renewable Energy. And broadly, I think with the time scales, every bit of these definitions and based on how you are going to project has been changing. And effort to decarbonization, I gave you an example very, very clearly saying that electricity is being really supported through wind and solar. That's major activity going on. Second one, I think you must have been seeing quite a bit, people coming from biotech must be very, very conversant with ethanol. People have been talking about ethanol. Ethanol is being really promoted quite a bit. 20% of ethanol is getting blended into the gasoline now. I think there's one way to see whether we will be able to offset fossil fuel for some reason, offset the emissions also for some reason. If the ethanol is coming from the so-called bio route, I think most of the ethanol today is coming through sugarcane route, still sugar biomass is CO2 neutral. We'll come back to that question very clearly in a slide or two. But broadly speaking, I think this is the contour on which it's being done. And various other countries, for example, Brazil has moved significantly into the so-called flexi fuels. They generate ethanol and put ethanol and gasoline together. They have developed engines which can also be flexi engines. I think that's the way things are happening in Brazil, but India is also catching up on those aspects. And bio-CNG, you must be aware of, any fermentation process under controlled conditions, anaerobic conditions, you can generate methane. And that it's called biogas generally in the conventional term. It's a typical simple process which takes place. And once you remove the CO2, this will be comparable to the CNG what we are importing. That is why the prefix with bio gives it the tag saying that it is a renewable, green, can it be sustainable? These questions can be answered. But broadly speaking, this is how it gets connected to the broad energy scenario and still far ahead from what needs to be done. There are various programs which have been set up to deal with this one. Many of these things are going on in some way or the other, something to reduce the CO2 footprint, which can be really looked at. There are various other things which I don't know, people must have heard of Griha rating, where you want to really look at buildings. People have been really looking at coding in such a way that Can I reduce the amount of energy I use? Can I reduce the refrigeration load by using some air circulation and ventilation through certain, the way I position the building? Can I bring in more air naturally? I think these are some things which are happening. I think to that extent it has an impact, but does it create a big impact? No, not lot more has to be done. Just to give you a glimpse, most of you would have read this one. I thought I should just give you, when you have a solar in terms of radiation, it contains various wavelengths and other things. Let us not get into the science. Based on a panel, the electrons generate the voltage, and you go through a storage called battery. You can use it the way you want. This is what conventionally, even if you really look at a small household system to large megawatt level, this is the process which is, and this has gained quite a bit of popularity in the country, and now we have gigawatts worth of such things happening. Unfortunately, we don't have basically major manufacturing facilities in India. Instead of importing oil, we are now importing the panels from China. I think that's something, whether it's sustainable or not, I think you people can now start thinking along the verticals what we spoke about. The other major impact, I think this part, I think most of you would have seen now using solar collector. This is being promoted quite a bit, and many of these, it's also becoming a norm in terms of rules. How do you really look at these as a standard package, either in a society, that means in a apartment or in a given location or for an industrial use? I think these are becoming standard, and also has an impact with respect to the necessary, I should say, rules and regulation kind of thing. People now have to bring in the so-called solar water heater and solar PV as a part of certain square foot area beyond that, just to make sure that you will be comparable or compatible with the kind of norms what is required. The other major thing which is required, which we have not been able to do too much, is with respect to the solar thermal. There are a few other things wherein we can capture the energy in the form of heat and use the heat. One such thing we have been able to do for water, but there are other applications wherein you should be able to run megawatt power plants using this heat. It's called heliostats and other things. It was becoming too technical. I thought it's not relevant right now, but you can see this is the rate at which we have been growing. Over the last 10 years, the rate at which solar has picked up is significant. About, I think we have been able to go much closer to what is there. This is the published information. I thought that would be useful for you. I think we have been able to do that much more, and now the leverage is to make sure that how do we store beyond this one. For example, if you want 24/7, batteries become very, very critical. That's how you really look at what needs to be done. If you really look at, this is the time wherein we can think, how do we really look at sustainability? The word sustainability had long term. Does it have an impact? What does it happen? See, most of the work which I mentioned to you has been renewable, has been spoken about over the last 3-4 decades. While the products have been developed, whether it has reached a stage wherein we go through a condition where there is going to be a trash. That means at the end of the day, there is going to be so much of material. For example, if we take a solar panel, it is made of multi-layer, I should say, metal matrix which are there, right from aluminum to high rare earth materials to steel. Everything has been put together. I think somewhere, about 90 tons of material is going to be there per megawatt of the power. Roughly, this is the kind of thing which is there. Now, there is a question people are asking. Let us say that life of a panel, let us say 20-25 years. Here they have done that one. If it becomes slightly earlier than that, what is the impact? Early so-called replacement, which you really looked at, it can make a very, very big. Is there a facility wherein you will be able, today we have been looking at, if you discard a car, is there a place wherein we can really take it away to get into scrap? It's not so in India still. We have to go a much, much larger time scales to deal with that one. Slowly it is happening, but that's not getting implemented the rate at which it should happen. I think these are the questions which people have been really looking at, what needs to be done, how do we take care. Just to give you some glimpse about what needs to be done, when you really look at coal-based power generation, we are looking at about 1,000 grams of CO2 for every kilowatt of electricity what you generate. Now, we have been talking about can I use solar? Now you can see the difference between these two. This is where people have been wanting to see whether we will be able to reduce the carbon footprint by using solar. Whether it does the kind of duty cycle what the coal power plant does, no, it doesn't do. But still, it can really reduce your carbon footprint. If reduction is the carbon footprint is our prime reason, probably this is fine. Sustainability is the thing. Maybe it has some issues with respect to sustainability. If we want to really look at can it become energy secure, I think this can be, because it's in your boundary area, it's within your country. You can do the way you want, you don't have to depend on somebody else. I think these are some questions which will become very critical. Similarly, when you really look at wind, simply it just says that the rate at which the wind speed moves, there is a particular limitations on which you can do. You can generate about 1/3 the power what the wind carries through, and that's called Betts limit. And based on the windmill, you have the necessary generator which is connected to a controller which maintains the speed, and you can generate electricity which can go either to a battery or to the grid. I think all these options, you must have seen these several windmills these days, right from it started off with a few tens of kilowatts. Now we are having megawatt level wind turbine. That means as you go higher and higher, you can have a very large wind blades. I think this the dictating term would be the radius of this. Bigger this one, larger is your throughput, and it has its own issues with respect to so-called the weight balancing and various wind loads and other things. I think these are now slowly getting developed. In fact, initially when we started putting these technologies about 15, 20, 25 years back, our controller it's a very, very important component. Depending upon the speed it rotates, it will either overspeed it will control, underspeed it will also control. I think keeping that in mind, this controller becomes important. At one point of time when we started implementing where we didn't have the necessary technologies and controller, probably it used to draw electricity from the grid and operate like a fan. These were the initial days when during the 80s kind of situation. Today I think the technology has matured enough, and majority of the hardware is still done in India now, and some of those important components with respect to motors and other things, certain things still gearbox do come from abroad. This is something which you need to contrast with respect to low life cycle energy. As long as the major thing is the impact with respect to the metal and the concrete what you put in for the entire structure, beyond that there is nothing. Your overall emissions is going to be much lower, and this is something which none of us would have imagined. In California there is a big law which says that because the noise level is so high that birds cannot exist in mill areas, there are laws which will prevent people putting wind farms in certain sectors. I think many of these things are becoming poor sighting causes most conflict, not wind itself. I think this is the issue which I thought I should just visual and noise concerns near habitation. I think these are all becoming very, very serious concern. Hydro, I thought I should just give you a glimpse. What is done in hydro, you would have seen most of these things. There is a reservoir, you create a pressure head, and based on the pressure head you get the velocity to go through the turbine here, which will generate electricity. This turbine could be any of these various classes of turbine which is going to be there. And if you had an occasion to see that, there is a huge catchment area, then you have another place, it's called a dam. From the dam you will have various maybe stages under which it will come through a penstock, it will come into the thing, and the tail race that after extracting the energy it can go for other purposes. This is how typically, and if you have very large thing, the amount of catchment area what you need is going to be very high. This probably is not sustainable. That's the reason why large hydro is not even called as green, because so much of both habitation and the people surrounding will not be able to stay there, and we are going to lose a lot of area that doesn't contribute towards the thing. And this is where some of the additional insights into this. Very low operational emissions. This is extremely low because life cycles emissions can be very, very low except for what you have installed. And the ecological damages could be river fragmentation, the way you are going to divert the entire stream. This could impact, and a range of things related to this is what is being looked at. And the sustainability component will really look at runoff of river, small, medium, and pump storage. Okay, this is a very, very important if people are following on the energy aspect. If electricity is generated from wind or solar, one way to do that is to put a battery like any of our mobile phone will have a battery. Similarly, you can have large scale batteries. The UPS is one great example for that one. And these are, people have been looking at industrial batteries. But there is another option for you. Wherever there is going to be a water, you can pump water from the ground level using the surplus electricity, take it to elevated level, and whenever you want, you can run the turbine. I think that becomes, this is called pump storage. This is becoming slowly famous. The context has been in terms of, if you want a 24/7 green electricity, which I'll come to that definition later on, this is one option people are exploring. That means if you have an elevation, you have water which can be recycled again. You bring it to the bottom and pump it back. I think your evaporation loss is all what you need to compensate kind of situation. I think these things give you a broad glimpse about how do we really look at various energy sources which are commonly known to us, but people looking into your biotech and other things, you are familiar with the terminology called biomass, because it's also a so-called, it's a carbon, hydrogen and oxygen matrix which is there, which is a plant growth. I think by definition, biomass itself could be plant or animal matter. Broadly speaking, if you go through the Oxford Dictionary definition, it is plant or animal matter, dead matter. In our case, we can consider probably the plant matter. And biomass is also called as a CO2 neutral technology. What do I mean by that one? See, for example, if you have 1 kg of fuel, if you, let us say this plant has generated 1 kg, you use either in a chula or in a power generation, that carbon which is going to be here will get converted to CO2. That CO2 can be fixed by photosynthesis again. If you can do that one, then it is a CO2 neutral technology. That means you are not putting more CO2 into the system, unlike the fossil fuel, wherein at the rate at which you are utilizing is no comparison to rate at which it was being generated there. I think that is the mismatch being there. Here it is not so. And similarly, this is simply an example to show that the entire cycle can be a, if you go through CO2, through photosynthesis, you get this and it can go to a power plant. That means the entire cycle can be looked at as, this is what people have been looking at. Can we see that the carbon footprint can be reduced? People have been talking, for example, today electricity is generated through coal. We know that it emits about 1,000 grams of CO2 per kilowatt or megawatt hour kind of thing. Similarly, can we really look at this and try to see, here there is no impact with respect to that one. And I think most of you would be aware of this fermentation process. I'll go little into a bit of technology so that you will understand where we come from. This biochemical conversion process, you know that if you put a food waste just overnight, you'll start smelling. It's nothing but microbial activity taking place there, and whatever you had the starch, it goes through microbe, some conversion would take place and emits so-called some gases. Under controlled conditions, these gases can be really looked at, right from the way we convert our milk to curd is an example of fermentation process. You just bring in some microbes there, it does all the necessary job. But rate at which it happens is very, very slow. That I think we need to, if you really calculate the rate expressions for that, that's something which we need to be aware. It's going to be in terms of tens of days kind of thing which has to happen. This is the process which is there. This is what I said, bio-CNG process which is being supported to Government of India through various routes. You take any substrate, go through a fermentation process. It has all the sub-activities right from hydrolysis, acidogenesis, fermentation. I mean, all the processes do take place. Finally, you convert them into simple sugars, which will enable for the methanogens to generate methane, or you can do various other controlled processes to get ethanol or methanol. A range of things, whatever is required, can be tuned. I think that way biotechnology has really created a portfolio of downstream chemicals or fuels which can be generated based on how you can tune. Except that we need to be aware that depending upon the temperature, it could be either thermophilic or mesophilic. Either of them, it could be in that range, maybe at medium room temperature or high temperature. This thing, the rate at which high temperature, you can really bring it the time scales to much half. I think that's something which you know. And once you have the gas, there could be other processes. I'll come back to a little more with respect to so-called distribution of the feedstocks later on. There is another process called thermochemical. Here, biochemical was the word. Here, thermochemical. There, bio means it was mostly towards the bioorganisms which were trying to do the job there. Here, thermo means it's the heat which is also going to do the simple. Any of the chulas what we use is thermochemical conversion. You use heat, and the necessary chemistry takes place for you to generate heat there back. That means you take a biomass, you ignite it, or a matchstick, you ignite it. After the chemical is over, you see a flame. That is because of the thermochemical. There is heat and chemistry both happening. Because the temperatures are going to be larger, the rate process is very, very fast. If you take a grain and allow microbes to act, it may take 2 days. If you take a grain and put it into the fire, it takes seconds. This is as simple as that. It's that rate process. A is equal to E by RT. The temperature is the one which dictates everything for these processes. Now, when you do various under control processes, you can also generate a mix of gas which can be utilized for downstream processes. And what is actually biomass when we really look at as a chemistry part? It contains carbon, it contains oxygen, it contains hydrogen. It is a CHO complex. The same CHO complex, over thousands of years, what has happened is the O has got eliminated and you get the hydrocarbon, C and H. That's what we use. All the diesel or the gasoline is CH1.2 to 1.4. Roughly that is the kind of distribution. O is not there at all. There could be some sulfur and other things which come up because of the range of bioproducts which have gone into the fossilized conditions. The other beauty is the CHO complex doesn't vary too much on an ash-free basis. For example, if you take rice husk, you would have seen rice husk. Rice husk contains 20% ash. Nature has designed the grain in such a way that if you really look at why it has designed the husk the way it is, it will not get affected by any insects, or maybe the birds cannot eat the grain. The outside structure is so well structured, it is completely silica matrix and interspersed with lignin. Lignin is an extremely hard binder, I don't have to tell you. It's a great binder between cellulose and hemicellulose. That is the one which holds the grain. The moment you remove the husk, you can see it is extremely abrasive. Husk is very, very abrasive. It is basically silica. And if you make sure that rice husk contains typically 20% ash, on an ash-free basis, most of them will have CHO same. And whenever we are looking at the properties of these fuels, either diesel or something else, there is one very important component called calorific value, which you would have come across. on an ash-free basis, the calorific value of most of the fuel should be roughly same. I thought I will just leave this thought process in you. And sustainable needs, yes, when we really look at the total, if you want to really look at energy perspective in any of the goals here, wind needs to be better siting and circularity, solar needs decentralization, clean manufacturing, hydro. I think various other portfolios which needs to go along with that has been brought to make sure that whether we are going to be sustainable, whether you will be able to meet everything in the same manner is a difficult question, but I think these are the textbook requirements when we want to really look at this one. I'll just touch upon some work what we have been doing at IISc on biomass so that you will get a glimpse. I'll take another 5 to 8 minutes kind of situation. See, when we look at hydrogen, you know that all the hydrogen produced in this country or elsewhere, which is used for maybe refining, maybe people who are working in the food technology, they know that hydrogen is used for refining various things. You need hydrogenation process wherein you bring in hydrogen. Similarly, for all our oils, when they have to be desulfurized, you need hydrogen. If you want to go from Euro 4 to Euro 5 or BS4 to BS6, you need more hydrogen, because hydrogenation is the way you can remove the sulfur. And for generating the hydrogen, they take methane, subject it to what we call as steam methane reforming. They subject it to that and try to generate the hydrogen. Every kg of hydrogen you generate this route will put in about 10 kilograms of CO2. This is how currently in India we consume about 6 million tons of hydrogen every year. Basically it will go for refining. Second one, it goes for ammonia manufacturing and other industrial applications slightly. The major consumers are the fertilizer manufacturing and the so-called the refining. I think majorly it goes into these things. And we need to be aware that yes, if you are going through this route, you are going to produce this. This is where the National Green Hydrogen Mission has come into play. In the country, there is a big program going on to see whether we can generate hydrogen, and we have nomenclatured it as green hydrogen. I'll come back to that, why green and how can we define it to be green. And this is to see whether we can mitigate this one. This CO2 what is getting liberated here is a possibility. Now, I don't know how many of you have heard of carbon capture. Have you heard of this term called carbon CCUS, carbon capture utilization strategies? I think carbon capture is one way wherein you will be able to, if you can take the CO2 out, is one way, so that the product of hydrogen says that I have been able to take the CO2. Can we use it is the next question. How do you really? I think that has become a very big area of research. I think in the recent budget, if you go back, there is about 20,000 crores which has been apportioned for CCUS only, carbon capture utilization strategy. I think these are technology, and nothing is available globally which can be economical. It's extremely expensive. What can be done? One basic thing I'll tell you, the CO2 can be taken into calcium oxide, it can become calcium carbonate. I think these are some things which people are trying, but I think there is lot of things which as youngsters you can come with innovation. If you have some ideas how to play around with the chemistry to look at products, there's a great opportunity to come up and do something. And once you generate the CO2 H2, this is what happens in a typical refinery. When you go to petrochemical refinery, people talk about I generate ammonia, I generate alcohol, I generate waxes, I generate acetic acid, olefins. I think most of it, these terminologies are conversant to you. All this is coming through currently the so-called fossil fuel. You take a CH hydrocarbon, subject it to certain treatment, you get a mixture, and these mixtures go through a catalytic conversion route to get methanol is something which people are contemplating to be used in the shipping industry called maritime fuels. Because now the ships use heavy oil or diesel as the transport fuel. Now they would like to reduce the carbon footprint for that one. People have been looking at methanol. Similarly, people have been looking at, there is another component called sustained aviation fuel. For example, one of these fractions today when you really look at the aviation fuel, it's also one of the derivatives from the COH2 only. And people have been looking at, can I get that from green fuels? It's called SAF, Sustained Aviation Fuel. There's a big activity going on across the globe on this matter. And if you really look at why we have been looking at the so-called other than fossil fuel, there are only two naturally available substances which carry hydrogen as an element. One is water, that you know, that even from high school physics, you know, if you subject it to a electrical charge across two electrodes, you are able to break that strong molecule of H2O into H2 and O2. It's exactly that, and that in large scale is happening globally. Everybody is trying to look at what can be done, how do we deal with what needs to be done. These are the technologies with respect to electrolyzers, and the electricity what you use, roughly about 60 kilowatts of electricity is required for 1 kg of hydrogen to be produced. The electricity what you are going to use, if it comes from green source, then it is called green hydrogen. The standards which has been set is for every kg of hydrogen you produce, there we were generating 10 kgs of CO2 with methane as a fossil fuel. Here they have said that no more than 2 kgs of CO2. That means you should restrict your CO2 to less than 2 kgs, then you can qualify the entire process to be a green process which generates green hydrogen. I think that's something which is happening, and there are routes with which we can generate hydrogen from biochemical and thermochemical. I'll just concentrate the thing on the thermochemical. There is a process what we can really look at as a solid fuel. You take a wood piece or a corn cob, any such thing, coconut shell. Under controlled conditions, it can be converted into gaseous molecules. One, you know, through the biochemical route, you will be able to generate that one. This is through the thermochemical route, you can really look at species like carbon monoxide, hydrogen coming there. And that gas can go into a range of downstream processes called, it can go into a diesel engine to replace diesel. It can go to a gas engine to generate electricity at whatever capacity, so that you don't need fossil fuel support. And there are various combinations of this which has been put together, and we in India have about 250 million tons of biomass, and we, even with the kind of things, even if we take 10% utilization, we should be able to generate about 1.20% of our hydrogen today. There are various other calculations which can be really looked at. This is a simple chemistry. This is biomass. You do a magical reactor, you can generate about 150 grams of hydrogen for every kg of biomass. This is something theoretically that all the H from water can go in, all the H from biomass can go in, you will generate this. This is something which we have already commercially exploited for power generation. I will show you some examples of that. This is where we are. We can generate close to about 100 grams of hydrogen per kg of biomass. This is something very, very simple chemistry it looks like, but an extremely complex process when you want to go through a thing to generate so-called hydrogen from biomass. This is what we have done, and we also really looked into, we started out this work about a couple of decades back to see that can we make sure that if there is an oil crisis, our agricultural sector will not get seriously affected. That's how we started off this activity, to see that at a village level, can we make sure that power generation can be generated. I think we were very successful in doing that. We went through various things to generate electricity. We started giving electricity for drinking water and various other things. But somehow I think there are other implications by which I think we were not able to do that. The political system did not catch up. In the meantime, we really looked at large-scale power generation unit for megawatt level, which is commercial. It used to take coconut shell as a feedstock, generate about megawatt power. This is just to give you a glimpse, saying that to be proud that we developed this technology in India. We were able to give it to other countries like the US, Europe, Japan. I think there is a possibility to do if you carry out certain research in a certain format. This is agnostic fuel. We can take any of these fuel, generate a gas which can go to internal combustion engine. This is something which I mentioned, saying that technology executed south to north. This is something which happens. Yeah. And there are many plants what we try to put things together, and this is something which our technology has been implemented in Switzerland, Thailand, US, Japan, and various other countries. At one point of time we were saving about 50 tons of oil. Probably the technology is revisiting now, because if you have been looking at the environmental standards, probably the CBAM is something which people have been talking, carbon boundary assessment mechanisms, which become very, very important. And there they have to reduce the carbon footprint. Many of the industries are coming back to, in fact, we were trying to do biscuits. If you go to Parle or Britannia, they have 50 or 100-meter long kilns where they use diesel as a fuel. We have replaced the diesel with biomass-based gas. I think many of these things have led to making sure that we can generate green. And the impact, this is what I want to say, we were saying about 50 tons of oil. We were able to really look at 100 tons of mitigated CO2 kind of situation. And I think, unfortunately, the policies did not support biomass as much as what is being done today. I think to that extent, we have been really looking at this process. Whatever we generated with power, we are just eliminating nitrogen and generating a syngas which consists of about 50% hydrogen, about 15% carbon monoxide, some CO2, and because the CO2 is going to be CO2 neutral technology, you should be able to really look at this to be a green one. I think that is the challenge. And today in the lab we have about 5 kilograms of hydrogen being generated. We have operated fuel cells which can really operate, go to buses or a truck and various other things. We are in serious discussions to put them into bus and a refinery kind of situation, wherein you have developed everything. This is agnostics. You can take any of these fuels. They generate a mixture of gases. There is a separation process. We generate fuel cell. This is 99.99% pure hydrogen, which can go to a fuel cell. Similarly, we have tested fuel cells with the purities. It's going to be extremely high challenging job, but I think we have been able to achieve this one to make sure that you generate. This is the ultra pure hydrogen from Sillard, the biohydrogen generated in the lab. I think your performance is absolutely fine. We are exploring in terms of decarbonizing steel. You know that steel, whenever they get the ore, it is Fe plus O3 is there. They have to remove the O3 so that it becomes iron, and that is being done using carbon now. In the presence of carbon, you eliminate the O there. That is very, very hard to abate, very difficult, I should say, the so-called sector to abate. I think today we have been able to really look at that one, and we are trying to see whether we will be able to. This is something which probably I think most of you would have been looking at, the global warming potential for any biogenic CO2. What we talk about today as CO2, you know that the recycling time scales biogenic CO2 is, if you take 1 year rotation period, is global warming potential, which is defined as global warming potential of 1 kg of biogenic CO2 when compared to 1 kg of fossil fuel. Kind of situations what we can really look at. And with this, I thank all the people and groups which have been part of my journey into this one. And at the end today, we have been talking about, can it be a negative carbon footprint for biomass to hydrogen? I think that's where we are, and any questions, clarifications, would be very happy to come back.
Change is a world order. We cannot stop it. Everything keeps going on. But many human activities, they are hastening the process of naturally what is happening. That is the big problem. That's where the sustenance is. Because biological evolution took many years. Just if things happen in a short period of time, we will have more negative consequences than its.
I completely agree. See, when you have development, it is very difficult to put a barrier in terms of development. And see, we were looking at, yes, mobile phones will help. It does help, but at the end of the day, you also have to really look at how much power we are carrying. All of us, if you look at the battery storage, what we are carrying, it's not small. It's going to be, and similarly, whenever there has been a development, these energy, at the end of the day, energy becomes extremely critical. You will not be able to deal without energy. Today, if AI ML comes into picture, the amount of energy which is required is going to be humongous. It's not going to be easy. And at the end of the day, if you want to make sure it becomes through green source, it's going to be further challenges. And it's not, I mean, nobody knows how it's going to happen. Because electricity costs are going to be lower, that is the reason why Googles and other people are wanting to come to India and trying to set up. Whether it's going to happen or not, it's not going to be easy. But in the coming days, there are interesting opportunities for youngsters to explore new things which can happen, and there are also challenges which will come for each of those solutions to be penetrated. I think there are challenges coming from all angles too. See, no, that's how the current thinking is happening. I'll give you a simple example. Whenever we really look at an infrastructure, we are looking at, can it do? If you go to Bangalore airport, we thought that what we are going to build will last 15, 20 years. No, that's not true. I think to some extent, that's where the planning becomes extremely important, and we do not have resources for long-term planning. That is a major issue what we have today. If there is nothing, thank you very much. Thanks, Amma. Thank you.
Thank you, sir, for delivering such an informative and thought-provoking session. We are truly honored to have such a distinguished speaker amongst us. I request on behalf of MSRCAC and KSTA from Professor Jairaman sir, Vice Principal, MSRCAC, to give the appreciation letter to Professor Dasappa sir. I request the following students to meet Dr. Mukta and Dr. Saubhagya ma'am at the reception desk immediately. Mayura and Ashrita from Ramaya College, Nitin Raj and Akshay from Kristu Jayanti, Sri Lavanya and Indrani from Reva University. I repeat, Mayura, Ashrita from Ramaya College, Nitin and Akshay from Kristu Jayanti, Sri Lavanya and Indrani from Reva University.
special lecture series 2026 jointly organized by KST and MSRCSC Autonomous on Science and Technology for a Sustainable Future. I request the dignitaries to occupy the seats on the dais. Over the past 2 days, we have been enriched by insightful lectures, meaningful discussions, and valuable interactions, making this program a grand success. We are honored by the gracious presence of our esteemed dignitaries on the dais today. To formally welcome the gathering, I now invite Dr. Pushpa H., Principal, MS Ramaya College of Arts, Science and Commerce Autonomous, to welcome the gathering.
So good evening everyone. So today we have completed with the 2 days special lecture series on science and technology for sustainable future. So the session, the 2-day session, really it was really nice. I think all the students who have attended this particular session would have got a very good information. So now we have come to the end of this particular session, the valedictory function. On this occasion, I would like to welcome the KSTA chairman, Professor Raja Saab sir, and the eminent speaker of this particular session, Professor Dasappa sir, the vice principal of MS Ramaya College of Arts, Science and Commerce, Professor Jairama BS, and the CEO of KSTA, Dr. Anand R. On behalf of MS Ramaya College of Arts, Science and Commerce and KSTA, I would like to welcome all the delegates as well as the students onto this particular occasion. Thank you.
Thank you, ma'am, for your inspiring words and constant support. I now invite Professor S. Dasappa, Advisor, Interdisciplinary Center for Energy Research, Professor, Center for Sustainable Technology, IISc Bengaluru, to address the gathering.
Thank you. I think now more in terms of word of wisdom to share with you. Thanks a lot for this invite and being part of the entire journey here. I think now with the kind of I should say the transition which is taking place from the so-called what you go through your educational system and taking forward your life. Some people may get into further education through PhD programs and other things. I wish you all the best for that one. But there could be many others who would like to explore their skill sets, what they have gained through this schooling here in this college, in this setup, to explore a few things which could be really out-of-box solutions also. Today you must have really looked into the way the job profiles are going to be there, quite demanding in terms of what is expected from each of you when you really get into an industrial house or the so-called research labs kind of situation. I think to that extent, whatever skills you have acquired on your fundamentals becomes very, very critical for you to take it forward. I think on that there is no second thoughts. But as we move forward, I think with the age digitization taking place across all sectors, probably I think you also should enable yourself into a little more, while the courses may really take care of all the needs to introduce these topics together, but exploring something beyond what the course could tell would pave way for what you can do something very different. In the current age of what we have been talking about, AI and ML, and to some extent it's all important for you to get, because most of you come from with the bio background, maybe it could be a range of streams which you are going to get specialized into. Probably I think the pathway for really there are exciting times to come in. There's no, and there are also challenges which will come in. I just posed a few, one or two, three problems when we were discussing, but there are umpteen number of problems when we really look at what needs to be done under the broad umbrella of sustainability itself. There are, I think they right from a fundamental science point of view, wherein you need to carry out certain scientific investigations to get into what you want to do as a future end goal, to applications. I think that's where your other skill sets comes in. I don't know, there must be several enabling mechanisms which is created by various institutions, including under the so-called schemes of government. Maybe I do not know how many of you are curious to get into startup mode. I think that's something which is becoming very, very important. And there are opportunities for you to group together, make sure that there are this idea which can inculcate during your college days and try to reap benefits with respect to that. I think there's something which is happening and happening at a large scale. Maybe the larger institutions are now, for example, IISc has been forced to look at into startups within itself. Similarly, maybe the IITs are doing that one. And being in Bangalore or in and around Karnataka, there is a possibility of many of you to use the skill sets, spend some time trying to see whether something innovative can be brought in, and there are umpteen number of opportunities. And all apart from your own institutions, there are other institutions which will help you in case you have something to deal with. If you don't understand a few things which is of then probably a next higher level, there are other institutions which definitely will be able to help you out. There are incubations, places wherein you will be able to explore your activities to see how you can reach that one. I think that is becoming the mantra of the day, trying to see whether you can become sustainable on yourself. That's something which you need to really explore, and there are opportunities. I think many of you would have come with certain grit to do this course, and yes, job opportunities are going to get restricted with the kind of things what is going to happen. And if the so-called AIML drives most of it service-oriented activity, probably I think that will pave way for something new to come in. I think these challenges are something which you need to keep in mind. And having said so, there are interesting problems to solve. That means there are avenues for you to get into professionalism in terms of dealing what you need to do that one. For you to get today the current status, I think the AI will provide you where we are, how you are, and what needs to be done, where you want to move forward. I think there are ways and means to deal with, to get a quick assessment of what needs to be done, either in your academic career, what you want to move forward, or an industrial career. It's something which I thought I should just share with you, and wish you all the best in your career pathway, and looking forward for further interactions with the group like this. Thank you very much.
Thank you, sir, for an enlightening and motivating address. We now move on to the felicitation ceremony, where we honor distinguished personalities who have significantly contributed to this lecture series. I request the dignitaries on the dais to kindly proceed with the felicitation of Professor S. Dasappa. Thank you, dignitaries. Now I request Dr. Pushpa H. Principal, Dr. Lakshmikant sir, and Dr. Prasanna Kumar sir to felicitate Dr. Raja Saheb, Chairman KSTA, former Vice Chancellor Tumkur University, and Dr. Anand R, CEO KSTA. series, I now invite Dr. Lakshmikant, Associate Professor, Department of Biotechnology, to present the report of the program.
Good evening. My sincere pranam to the dignitaries on the dais, off the dais, esteemed members of the KSTA, respected colleagues, and dear students. So it's my privilege to present the report on the two-day special lecture series on science and technology for a sustainable future. For this program, 178 students have registered, and the program commenced with the Nadagite and the KSTA song. And as per the schedule, the first invited lecture was delivered by Dr. R. Saudamini, Professor, National Center for Biological Sciences, on computational design of a novel dengue vaccine focusing on eliminating antibody-dependent enhancement. And at 11:30 a.m., a formal inauguration function was conducted, and Dr. Rajasab A.H., Chairman, KSTA, and former Vice Chancellor, Tumkur University, warmly welcomed the dignitaries and the gathering. And Dr. Pushpa H., Principal, MSRCAC, gave an overview of the special lecture series. And this program was formally inaugurated by Dr. M.R. Sitaram, Honorable Chairman, Gokula Education Foundation, and presently member of the Legislative Council, Government of Karnataka, and former Minister for Planning, Statistics, Science and Technology, Government of Karnataka. The guests of honor were Dr. Parshanath H. V., IAS retired, former member KPFC, and Chief Executive, Gokula Education Foundation, and Dr. Karisiddappa, former Vice Chancellor, VTU, and Chief Academic Advisor, GF, and Sri Ramachandra Ji, Chief of Finance, GF. Dr. M.R. Sitaram sir was felicitated by the dignitaries, and in his inaugural address, sir emphasized the importance of science and technology and encouraged the youngsters to inculcate curiosity and a research mindset, and highlighted the role of education and youth in nation building. Later, Professor Rajasab H., Chairman KSTA, gave the presidential address, followed by the vote of thanks by Dr. Anand R., CEO KSTA. After the inauguration session, the second invited lecture was delivered by Dr. Chandrasekhar M. Biradar, Earth System Scientist and Lead, Global Green Growth, and Founder and CMD, Global Green Growth Corporation. And his talk was on augmented science technology and academia for restoring regenerative future. The third invited lecture was presented by Dr. Raghavendra Ji, Assistant Professor, University of Horticulture Sciences, Bagalkot, on genome editing and sustainable agriculture, role of CRISPR in food security. And on the day that is today, the fourth invited lecture was delivered by Dr. K. Shusha, Scientist C, National Institute of Virology, Bangalore Unit, Bangalore. And Madam spoke on emerging infectious diseases, virological threats, and preparedness for the future. And the fifth invited lecture was presented by Dr. Meenakshi Kamraj, DST Inspire Faculty, Department of Bioengineering, Indian Institute of Science, on advanced biomaterial technologies for tissue regeneration using 3D bioprinting. And based on these lectures, a quiz competition was conducted, and which were delivered from the eminent resource persons from various disciplines. And the sixth invited lecture was delivered by Professor S. Dasappa, sir, Advisor, Interdisciplinary Center for Energy Research, and Professor, Center for Sustainable Technologies, IAC. Sir spoke on the renewable and green energy technologies for a sustainable future. And and sir is also the chief guest for our valedictory function. With this, I conclude the report, and thank you.
Thank you, sir, for the detailed and informative report. I now invite Dr. Raja Saheb AH, Chairman KSTA, to deliver the presidential remarks. Learning is complete only when reflections are shared. At this moment, I invite a few participants to share their feedback and experiences about the lecture series.
Good evening, everyone. I take immense pleasure in sharing few words of feedback about this whole session. And first of all, I would like to thank our college, MS Ramaya College of Arts and Science and Commerce, for organizing this event. It was really amazing. And all the speakers who had come for this session, they had really strong like the subject knowledge, and all the all the topics that were discussed here, like emerging virus diseases, viral diseases, and 3D bioprinting, they were really interesting. And all the sessions were really well placed, and it was really interesting. And the fun part was the quiz, which was organized based on the session. And as sir was saying, there are real problems which is going around the world, and with our knowledge, we would be able to, get a solution for that. And overall, the session was really good, and I really look forward to attending many more similar sessions like this. Thank you so much.
Is anyone from other college would like to share the experience? Kristu Jayanti or Eva University, anyone?
Good evening, everyone. I'm Pooja K from Reva University. I'm currently pursuing my bachelor's degree in microbiology, chemistry, and genetics. I'm very honored to be attending these lecture series organized by Ramaya University in association with KSTA. The two-day lecture series was very, very informative, and I'm honored to have listened to various eminent speakers from various esteemed institutions. And my personal favorite session was about virology, as it is very relevant to what I'm studying, and I've developed an interest for virology since COVID. Overall, the event was very well organized, and thank you.
Thank you, Akshita and Pooja, for valuable feedback. Now we'll move to the next session of the program, that is certificate distribution. I request one representative from each college to come and collect the certificate. First, from Eva University, please one of you come to collect certificate. I also request the dignitaries to kindly proceed with the certificate distribution. Yes, from Reva University, any one student can come and collect the certificates on behalf of all participants. Next, Kristu Jayanti can be ready. Certificates. Participation. No, no, sir. Participant certificate. Come on this stage. Come on this stage. Kristu Jayanti. Any student from Kristu Jayanti? Ramaya College students. Ramaya College student, each department, CRs be ready. First BT 4th semester, Biotech 4th semester, CR. Followed by MB department, 4th sem CR. Chemistry, Biochemistry, CRs. Second semester follows the same order, BT, MB, then Biochemistry, CRs. Kindly come on the stage and collect the certificates of your classmates. They are ready. Time it is. A huge round of applause for our media students who powered through 2 days of intense non-support recording and production to bring this lecture series to life. I request Fardeen Ali and his team to join us on the stage to receive your certificates. Please come on the stage. The whole team can come. Yeah, okay, one. Thank you, dignitaries. We now move on to the one of the most awaited segments of the program, that is prize distribution. I now request Dr. Prasanna Kumar, COE, to kindly announce the name of the winners and also request the dignitaries to proceed with the distribution of prizes.
So, good evening, one and all present here. So, actually, now come to the announcement of the results of the quiz competition. So, we appreciate the enthusiasm and knowledge displayed by all the participants. So, whoever wins, so congratulations to all the achievers, and we look forward to celebrating your success. So, totally 23 teams are participated. So, from the different colleges. So, Kstacademy, Reva, MSR, Anupatanga University, and JSS. So, total in all the teams, so 3 teams scored same marks. So, that means a tie. So, because it happened, so that we conducted again the quiz. So, in that whoever giving the answer within the less time, so there we taken as winners. So, after asking the questions and doing again the quiz, so as per the time allotment, so first third team taken 2 minutes 5 seconds to answer the questions what we asked, and second team taken 1 minute 52 seconds, and the first team taken 1 minute 38 seconds. So with the close thing, it happened, but all the students really the participation and whatever the knowledge they gained from these 5 days, 5 sessions, so very appreciable. So now I'll announce the college name and the winners. So I request the dignitaries to award the prizes. So third prize goes to Kstacademy College, Nitin Raj and Akshay. The team Nitin Raj and Akshay. faculty students. The team are Sri Lavanya and Indrani S. And first prize goes to Mayura and Ashita from MSRCAC. So congratulations to all the winners, and a special appreciation from the academy to the media team because of their continuous effort and coverage of these 2 days sessions from the academy. So I request the students of journalism of MSRC to take the prizes. Now I request our Mohammad Fardeen Ali. Rahul Sarup. Fardeen, come. So appreciation from the KSTA team. Rahul, Bhumi Bakshi, Anushka Chandra, and Ananya. So now I request Dr. Rajasab A.H., Chairman, KSTA, so to give presidential remarks on this valedictory session, please.
Respected dignitaries on the dais and off the dais, Dr. Dasappa, then Dr. Pushpa Madam, Principal of Ramaya Arts, Science and Commerce College, then Dr. Anand, CEO, then our Vice Principal of Ramaya Arts, Science and Commerce College, Dr. Savita, Dr. Srinivas, then Mahadeva Gowda, and our senior Shri Dr. Chandrayappa. He is 2 PhD, double PhD, and all others, the Lakshmikant and others who have organized this conference. And you have done extremely well in organizing the conference, especially the staff of Ramaya Science and Commerce College, as well as staff of the academy. They worked very hard to make this a reality, 2 days lecture series. And then they chose very eminent personalities to give lectures on emerging topics. Each one of the topics they chose is of a emerging importance in the present scenario. This is what we needed presently, because we have to expose our students for the upcoming branches in the science, and also such of those subjects which are applicable to the society in reality. So the period of descriptive science is over. Now it is only the period of applied science. Whatever knowledge we have gained, whatever expertise we have gained, it has to go to the common man and for the overall development of the society, thereby adding development of our nation. That is why we brought in the word sustainability. One of the first speakers, Dr. Chandrasekhar Biradar, was telling, we have to sustain, we have to sustain, we have to sustain future sustainability, like that we use the word. Already we have lost the balance in maintaining the sustainability. That is the crux of the problem. When we lose the balance, we try to attain the balance and maintain the balance. That's what is sustainability is at present. It is at stake. Whether it is food production or health or economics or welfare of the society, everywhere there are signs of inequality. And then threats. That's why all of us have to lead our life in such a way that we do not disturb, we shall not disturb the balance in the nature. It can be at the micro level or at the macro level or at social level, economic level. So many all things are brought in here in this maintenance of sustainability. Science and technology also plays a vital role in maintaining sustainability, because without that society cannot be healthy. There are various branches of science which aim for this and working for this. The first and foremost duty of the citizens of India is to bring in equality. Equality in standard of life, equality in education, equality in output, and equality in performance, and overall success of each and every citizen of the country. Then only these 17 goals of sustainability can be achieved. That's why we were very particular to put the 17 goals. 17 goals. Sustainability involves 17 goals there. If you have not read them, take photograph of them if you are not having. And it is there in your colleges also. I am sure that in Ramaya group of institutions, every college has a board, sustainable goals. Similarly, those who have come from other colleges, institutes, you should tell your administrator to raise the board of 17 sustainable goals. Every day you should see them and behave yourself, including the scientists, professors, all the people. Then our consciousness will become little more refined, and we become more sensitive. I am also responsible for maintaining the sustainability in my way of life, including teaching, research, or whatever it is. Thinking, behavior, all these things become important. And in science, for example, there are emerging fields nowadays. During our days, we were only understanding nature by way of description. We used to understand what are bryophytes, what are pteridophytes, what are higher plants, how molecular reactions take place, what is the genetic makeup of different organisms. We studied classical science in different fields, including biotechnology. Now, AI technology has come, quantum technology has come, precision agriculture has come, and even in language, for example, translating the scientific discoveries, innovations, skill development in local language is also very important. For example, in Karnataka, science has to be brought in local language also. Unless you bring science in local language, your colleges will not sustain themselves, because sometimes they will have to be taught in their local language at village level or panchayat level, taluka level, at school level. One has to teach in their mother tongue. It may be, in our case, Kannada. In other cases, it may be different. So science has to be popularized, and also it has to be understood through their mother tongue. So that also becomes very important. For example, in quantum technology, probably in next 5 to 10 years, Professor Dasappa, maybe they are more into the subject here in University of Science. This quantum technology, they say that earlier only physics people used to study quantum mechanics. Now everybody is putting quantum, bringing quantum into their subjects, because it is applied in agriculture, it is applied in space technology, it is applied in health science also, in diagnostics. Especially there is one aspect of particle entanglement. I have not understood it completely, but I heard experts speaking on particle entanglement. If one particle is on Earth, another particle is in Mars, they entangled with one another. Using this philosophy, they are using the very precise diagnostic in medical sciences without harming the biological systems, without invasive investigations. Just by quantum technology, it is possible. It is possible to diagnose the diseases or abnormalities in the human beings. So using AI technology, yesterday I was telling you, it is possible to analyze the soil structure, soil composition, concentration of various macro and micro elements, what is to be done to that soil at different places, and they recommend a dosage of application of fertilizers or micronutrients or macronutrients or biologicals. This is one aspect only. Similarly, drone technology. Drone technology is used in space, it is used in defense, it is used in agriculture, it is used in medicine also. For example, remote villages, tribal areas, they are suffering from disease. Hospitals are not there, telemedicine. Okay, then medicines are not there. What to do? Send the medicine through the drones to that area. Like this, applications are innumerable in the field of science, especially in AI as well as quantum and precision agriculture. Just I give one example. Professor is expert in that. In precision agriculture, you will grow the plants both outside as well as under controlled conditions, both indoor and outdoor conditions, and then temperature, relative humidity, and other ambient air qualities will be maintained. Whatever fertilizer is needed is given to the plants without wasting the fertilizers, without wasting the chemicals, or without wasting the fungicides, whatever it may be. So what is needed by the plant is exactly supplied to them. And vertical growth is also possible in agriculture. All these years there was horticulture, horizontal growth. Now vertical also it is possible. In a given space you can grow the plants horizontally as well as vertically. So Japan was the first country to practice this, because they have very less agricultural land. They did this. And then this precision agriculture, now marine agriculture. Imagine, whatever we have on the surface of the earth, land, we also have lot many good things in the inside the sea. Food plants are there, and then fishes are there, and corals are there. So many plants with medicinal qualities are there. Sea is a hidden treasure in all respects. And using our technology, it is possible to investigate the treasure of the underground sea. So these are the new openings in the various aspects of science. And then maybe next, maybe sorry, in April, if our CEO and other staff agree with this, we can have one conference on critical minerals, that is rare earth and all that. Critical minerals or rare elements. It is for the rare elements now the world crisis is created. If you read newspapers, you will understand. In Ladakh area, Jammu and Kashmir, Ladakh area, it seems it has very rich minerals. They are necessary for our mobile technology and also for rare chemicals used in space, used in aircrafts, and used in the warfare. These rare elements are critical elements. That's why China has occupied some parts of Ladakh already. Okay. Similarly, our great US President, what is that? Donald Trump, he is asking that I want Greenland, right? I want Greenland. Every day he says, I want Greenland. Is it a chocolate to give? Children usually in the house, they ask, Mommy, I want this chocolate, chocolate, chocolate. He has gone to that extent. And once I heard him talking, if Greenland is not given to America, there may be a war. War. So if America takes Greenland, there will not be world war. There is no world war. If Greenland is given to him, which does not belong to America, then America will see that the whole world is in peace. Why Greenland is the question. I am not worried about that politics. Leave it. Why Greenland? Why Ladakh? It is because of the critical elements, critical minerals, actually. So yesterday one expert came here, Dr. Sangurmat. He was director of the Hattigold mines. He is expert in critical minerals. Both central government and state government is taking his consultation, actually. His article came in Deccan Herald also. So I have requested him from the academy side, why can't we have a brainstorming meet or lecture series or conference or workshop, whatever it may be, on the importance of critical minerals. This is what I am thinking. We are thinking rather. Second point is virology. Madam was telling, and one of our students from Reva University, I think, Reva, she was mentioning she is a virology student interested in virology. This is the emerging field actually. Virology is the most emerging field in most of our research systems. For example, the other day, about 4 or 5 days before, I was in Nagpur in a conference to give a keynote address. There one of the farmers asked me, in Nagpur, soybean is the major crop. As we use our this sunflower oil, groundnut oil for our cooking here, in Nagpur they use soybean oil for cooking. It's a major oilseed crop. So he asked me, soybean is suffering from disease. What is the disease, he asked me. I asked him, please show me photographs. He showed me photographs. It was a viral disease. Later plants turn black. Why I am giving this example is like this, hundreds of examples are there in different parts of India and other parts of the world. These viral diseases are not easily controllable. Whether viral diseases of human beings, animals, or plants, it is same. There is no any we have antibiotic for bacteria. We have antifungal antibiotics for fungi. But what chemical we have for viruses? Nothing. Nothing. For the pandemic which we badly experienced, it is only vaccine which has saved many of us. And for that vaccine discovery also there is a story. Bharat Biotech, which is there in Hyderabad, Bharat Biotech, it within 6 months, usually vaccine in earlier days used to take 10 years to develop one new vaccine, 10 years, and lately at least 5 years. Other vaccines, to discover a vaccine or to develop a vaccine, minimum 5-year period is needed. But Indians, Bharat Biotech brought out vaccine within 6 months. That is the greatness of Indian minds. Greatness. That means if the opportunity is given, everyone will contribute to their best. Our minds are best. Only thing is paucity of opportunities, paucity of various other facilities, paucity of encouragement. That is how our really talented youngsters are suppressed. For example, some of the girls here who spoke, gave their their opinion. They speak better English than me, myself. See, that means they are better. If correctly, if they develop their future, they will be better than us, the younger generation. So India developed vaccine. You may not believe, one of the consultant for the vaccine development by Bharat Biotech was a chemistry student from Gulbarga University. Can you imagine? And he is coming to, we are inviting him to give a lecture on vaccines. One PhD from Gulbarga. Gulbarga, you all people, Bangalore people say backward, backward, backward. Region may be backward, economically may be backward, but every one of them is better than us. Every one of their IQ is excellent. I have seen them. Like that, no one is inferior, no one is superior. Only opportunities. Opportunities. We have to provide good opportunities for our youngsters. This is just an example. If possible, in coming year, we will concentrate or organize one good conference on virology also. How to control viral diseases? Savita madam may be knowing. Only vector management. Viruses are spread either by mechanical touching or transport or through the vectors. If you control vector, viruses will come down. But once virus get into the plant, that plant finish, Shraddha Madhu. You cannot recover. So, we are able to manage bacterial diseases. We are able to manage fungal diseases. We are not able to manage viral diseases. That's why more discussion on viral disease is also important. And one professor has given lecture on CRISPR gene technology. Unfortunately, I was not able to attend that. I was more interested in CRISPR gene. That is going to make a revolution in coming days. CRISPR gene technology. Those who are molecular biology students, biotechnology students, plant breeding students, they know what is CRISPR gene technology. Gene editing is possible from one species to another species. Required genes can be removed and then transferred, or you can delete them also, add them also. So that is another emerging technology. So like this, science has no limits. And I really appreciate and then congratulate you because you chose to come here for this good lecture series. And another matter to compliment you is, you are in Bangalore. If you travel to North India or outside India, except Chennai, Hyderabad, Pune, to some extent Mysore, Bangalore, except these three or four cities, you go and see other cities, you think you are 50 years behind Bangalore. Other cities. We will make out a difference there between Bangalore. Only the problem in Bangalore is the traffic movement. But for that, all other things are excellent in Bangalore. So feel yourself fortunate. Don't waste your time. Don't overdo AA also. AA is equally not useful. useful, equally not useful. Why I am telling this is to understand a subject, basic aspect of a subject. AI is okay. Within short time you will know history of that discovery or science or research. But if you want to do new research, AI is not going to help you. Your critical thinking, your keen observation, interpretational way of presentation, your originality counts, nothing else. Critical thinking, originality, scientific way of interpretation, your AI will not help you. If you depend on AI, completely on AI, even if you are a brilliant, then in the span of 2 or 3 years, you will become ordinary student. So use AI judiciously. That is my advice. Judiciously, without compromising your original observation, original thinking, critical observation. Close, you have to observe based on your background experience and understand the problems of the people. People, both rural and urban, understand how scientifically, how can I do better research and help them through technology. Then you get new ideas. With these few words, I stop here. I really appreciate Ramaya College of Arts, Science and Commerce, the dynamic principal, Dr. Pushpa, and also the wonderful team which is behind her. Wonderful team. I I, I cannot take name of all of you, but I can only say all of you are one, and you are working like family members to make the program success. That's why they are institutional members also of our KSTA. They have a special privilege to take the facilities of KSTA. With these few words, I thank all the professors and doctors who gave wonderful presentations here. I also thank all of you, and I compliment and appreciate those who have won the prizes 1, 2, 3, which you have made 1.228 second, 1.48 second, 1.2 what is that? 2 some second. That is not justifiable in my view. It is only to eliminate it may be helpful, but all of them are one and same in my view. Okay. So I congratulate all of you, those who have participated in the lecture series, those who have won some prizes also, and you must have made some new friends. That is also important. Keep the link and then friendship, and take help from one another. Try to develop your personality. You should become young scientists tomorrow. Thank you.
Thank you, sir, for your thoughtful and enlightening words. As we come towards the conclusion of today's program, I invite Dr. Prasanna Kumar, SG, Control of Examination and Associate Professor, Department of Chemistry and Biochemistry, to deliver the vote of thanks.
So, good evening everyone present here. So, ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಸೊ ವೇದಿಕೆ ಮೇಲೆ ಉಪಸ್ಥಿತರಿರುವ ಎಲ್ಲ ಗಣ್ಯರಿಗೆ ಹಾಗೂ ವೇದಿಕೆಯ ಕೆಳಗಡೆ ಆಸೀನರಾಗಿರುವ ಎಲ್ಲ ಗಣ್ಯರಿಗೆ ಶುಭ ಸಂಜೆ. MS ರಾಮಯ್ಯ ಕಲಾ ವಿಜ್ಞಾನ ಮತ್ತು ವಾಣಿಜ್ಯ ಮಹಾವಿದ್ಯಾಲಯ ಹಾಗೂ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಇಲಾಖೆ, ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ, ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಇಲಾಖೆ, ಕರ್ನಾಟಕ ಸರ್ಕಾರ ಇವರ ಸಹಯೋಗದಲ್ಲಿ ಆಯೋಜಿಸಲಾದ ಸ್ಥಿರ ಭವಿಷ್ಯಕ್ಕಾಗಿ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ವಿಷಯದ 2 ದಿನಗಳ ವಿಶೇಷ ಉಪನ್ಯಾಸ ಸರಣಿಯ ಸಮಾರೋಪ ಸಮಾರಂಭದಲ್ಲಿ ಈ ವಂದನಾ ಭಾಷಣವನ್ನು ಸಲ್ಲಿಸುವ ಗೌರವ ನನಗೆ ದೊರೆತಿರುವುದು ನನಗೆ ಅತ್ಯಂತ ಹೆಮ್ಮೆಯ ವಿಷಯವಾಗಿದೆ. ಧನ್ಯವಾದ ಎನ್ನುವುದು ಕಾಣಿಸುವುದಿಲ್ಲ, ಮುಟ್ಟಲು ಸಾಧ್ಯವಿಲ್ಲ, ಅದು ಹೃದಯದಿಂದ ಅನುಭವಿಸಬೇಕಾದ ಪ್ರಾರ್ಥನೆ. ಸೊ ಮೊದಲಿಗೆ ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಯಶಸ್ವಿಯಾಗಿ ನೆರವೇರಿಸಲು ನಮಗೆ ಶಕ್ತಿ ನೀಡಿದ ಪರಮಾತ್ಮನಿಗೆ ಮೊದಲು ನಮ್ಮ ಕೃತಜ್ಞತೆಯನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ನಮ್ಮ ಎಲ್ಲ ಶೈಕ್ಷಣಿಕ ಚಟುವಟಿಕೆಗಳಿಗೆ ಸದಾ ಬೆಂಬಲ ನೀಡುತ್ತಾ ಶ್ರೇಷ್ಠತೆಗೆ ನಮ್ಮನ್ನು ಪ್ರೇರೇಪಿಸಿರುವ ನಮ್ಮ ನಿರ್ವಹಣಾ ಮಂಡಳಿಗೆ ನಮ್ಮ ಧನ್ಯವಾದಗಳು. ನಮ್ಮ ಮಾನ್ಯ ಪ್ರಾಂಶುಪಾಲರು ಹಾಗೂ ವಿಜ್ಞಾನ ವಿಭಾಗದ ಅಧ್ಯಾಪಕರ ಪರವಾಗಿ ಗೋಕುಲ ಶಿಕ್ಷಣ ಪ್ರತಿಷ್ಠಾನದ ಗೌರವಾನ್ವಿತ ಅಧ್ಯಕ್ಷರಾದ ಆರ್ ಸೀತಾರಾಮ್ರವರಿಗೆ ಮತ್ತು ಗೌರವಾನ್ವಿತ ನಿರ್ದೇಶಕರಾದ ಶ್ರೀ ಆರ್ ಜಾನಕಿರಾಮ್ ಹಾಗೂ ಶ್ರೀ ಆರ್ ರಾಮಯ್ಯರವರಿಗೆ ಅವರ ಅನುಪಸ್ಥಿತಿಯಲ್ಲಿ ನಮ್ಮ ಧನ್ಯವಾದಗಳನ್ನು ಸಲ್ಲಿಸುತ್ತೇವೆ. ಇಂದಿನ ಸಮಾರಂಭದ ಮುಖ್ಯ ಅತಿಥಿಗಳಾದ ಪ್ರೊಫೆಸರ್ ಎಸ್ ದಾಸಪ್ಪ, ಸಲಹೆಗಾರರು ICAR ಮತ್ತು ಪ್ರಾಧ್ಯಾಪಕರು, ಸ್ಥಿರ ತಂತ್ರಜ್ಞಾನ ಕೇಂದ್ರ IISc ಬೆಂಗಳೂರು, ಅವರ ಗೌರವಾನ್ವಿತ ಉಪಸ್ಥಿತಿ ಮತ್ತು ವಿಜ್ಞಾನ ಕ್ಷೇತ್ರದಲ್ಲಿನ ಅಮೂಲ್ಯ ವಿಚಾರಗಳು ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮತ್ತಷ್ಟು ಅರ್ಥಪೂರ್ಣವಾಗಿಸಿವೆ. ಸರ್, ನಿಮ್ಮ ಅಮೂಲ್ಯವಾದ ಸಮಯವನ್ನು ನಮಗಾಗಿ ಮೀಸಲಿಟ್ಟಿದ್ದಕ್ಕಾಗಿ ಹೃತ್ಪೂರ್ವಕವಾಗಿ ಧನ್ಯವಾದಗಳು. ಡಾಕ್ಟರ್ H.V. ಪಾರ್ಶ್ವನಾಥ್, ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕರು, ಡಾಕ್ಟರ್ ಕರಸಿದ್ದಪ್ಪ, ಮುಖ್ಯ ಶೈಕ್ಷಣಿಕ ಸಲಹೆಗಾರರು ಹಾಗೂ ಶ್ರೀ G. ರಾಮಚಂದ್ರ, ಮುಖ್ಯ ಹಣಕಾಸು ಅಧಿಕಾರಿ, ಅವರ ಅನುಪಸ್ಥಿತಿಯಲ್ಲಿ ಅವರ ಮಾರ್ಗದರ್ಶನ ನಮಗೆ ಸದಾ ಬೆಂಬಲವಾಗಿರುತ್ತದೆ. ಅವರಿಗೂ ಕೂಡ ನನ್ನ ಕೃತಜ್ಞತೆಗಳನ್ನು ಸಲ್ಲಿಸುತ್ತೇನೆ. ಡಾಕ್ಟರ್ ರಾಜಾಸಾಬ್ AH, ಅಧ್ಯಕ್ಷರು KSTA ಹಾಗೂ PhD ಕಾರ್ಯಕ್ರಮ ಸಂಯೋಜಕರು, ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್, ಜನರಲ್ ಅಂಡ್ ಇಂಜಿನಿಯರಿಂಗ್ ಸೈನ್ಸಸ್, ಅವರು ಕೂಡ ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಉಪಸ್ಥಿತರಿದ್ದಾರೆ. ಅವರ ಶೈಕ್ಷಣಿಕ ಮತ್ತು ಅವರ ಸಂಶೋಧನಾ ಸಂಸ್ಕೃತಿಯನ್ನು ಉತ್ತೇಜಿಸುವಲ್ಲಿ ನೀಡುತ್ತಿರುವ ನಿರಂತರ ಪ್ರೋತ್ಸಾಹಕ್ಕಾಗಿ ಹಾಗೂ ಈ ಸಮಾರಂಭಕ್ಕೆ ಆಗಮಿಸಿದ್ದಕ್ಕಾಗಿ ಹೃತ್ಪೂರ್ವಕವಾದ ಧನ್ಯವಾದಗಳು. ಥ್ಯಾಂಕ್ ಯು. ನಮ್ಮ ಪ್ರಿಯ ಪ್ರಾಂಶುಪಾಲರಾದ ಡಾಕ್ಟರ್ ಪುಷ್ಪಾ H ಅವರ ದೃಢ ನಾಯಕತ್ವ, ಮಾರ್ಗದರ್ಶನ ಮತ್ತು ನಿರಂತರ ಬೆಂಬಲಕ್ಕಾಗಿ ಅವರಿಗೆ ನಾವು ಸದಾ ಋಣಿಯಾಗಿರುತ್ತೇವೆ. ಅವರಿಗೂ ಕೂಡ ವಂದನೆಗಳು ಸಲ್ಲಿಸುತ್ತೇವೆ. ಈ 2 ದಿನಗಳ ಉಪನ್ಯಾಸ ಸರಣಿಯನ್ನು ಯಶಸ್ವಿಯಾಗಿ ಆಯೋಜಿಸಲು ಅಮೂಲ್ಯ ಸಹಕಾರ ನೀಡಿದ KSTA ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕರಾದ ಡಾಕ್ಟರ್ ಆನಂದ್ R ಅವರಿಗೆ ನಮ್ಮ ಧನ್ಯವಾದಗಳು. ಹಾಗೂ ಅಕಾಡೆಮಿಯ ಸಿಬ್ಬಂದಿ ವರ್ಗ ಕೂಡ ನಮಗೆ ಎಲ್ಲ ರೀತಿಯಲ್ಲಿ ನಮಗೆ ಪ್ರೋತ್ಸಾಹ ನೀಡಿದೆ. ಸೊ ನಾನು ಈ ಸಂದರ್ಭದಲ್ಲಿ ನಮ್ಮ ಪ್ರಾಂಶುಪಾಲರಿಗೆ ಕೋರಿಕೊಳ್ಳೋದೇನೆಂದರೆ, ಎಲ್ಲರಿಗೂ ಸಸಿಯನ್ನು ನೀಡಿ ಗೌರವಿಸಬೇಕಾಗಿ ಪ್ರಾಂಶುಪಾಲರಲ್ಲಿ ಕೋರಿಕೊಳ್ಳುತ್ತೇನೆ. ಸೊ ಮೊದಲನೇದಾಗಿ ಶ್ರೀ ಶ್ರೀನಿವಾಸ್, ಹಿರಿಯ ವೈಜ್ಞಾನಿಕ ಅಧಿಕಾರಿ ಸರ್. ಸೊ ಚಪ್ಪಾಳೆಯೊಂದಿಗೆ ಅವರನ್ನು ಗೌರವಿಸಿ. ಉಮೇಶ್ ಕಟಗಿ ಅವರು, ವೈಜ್ಞಾನಿಕ ಅಧಿಕಾರಿಗಳು. ಮಹಾದೇವರವರು, ಅಡ್ಮಿನಿಸ್ಟ್ರೇಟಿವ್ ಆಫೀಸರ್. ದಿಲೀಪ್ ರವರು, ಆಫೀಸ್ ಸೂಪರ್ಡೆಂಟ್. ಶಾಂತರಾಜು, ಅಕೌಂಟ್ಸ್ ಡಿಪಾರ್ಟ್ಮೆಂಟ್. ಚಂದ್ರಶೇಖರ್ ಅವರು, ಉಮೇಶ್ ರವರು, ಪಾಂಡೂರವರು, ಲೀಲು ಮೇಡಮ್, ಮೋಹನ್ ಸರ್, ಭರತ್ ಅವರು, ಟೆಕ್ನಿಕಲ್. ಗೋಪಾಲ್ ರವರು, ರಮೇಶ್ ಅವರು, ಹಾಗೂ ಕೊನೆಯದಾಗಿ ಲಲಿತಮ್ಮರವರು. ಸೊ ಎಲ್ಲರಿಗೂ ಕಾಲೇಜಿನ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳು. ಸೊ ಉಪಪ್ರಾಂಶುಪಾಲರಾದ ಶ್ರೀ BS ಜಯರಾಮ್ ರವರು ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಉಪಸ್ಥಿತರಾದರೆ ಅವರಿಗೂ ಕೂಡ ವಂದನೆಗಳು. ಸಂಶೋಧನೆ ಮತ್ತು ಅಭಿವೃದ್ಧಿ ವಿಭಾಗದ ಮುಖ್ಯಸ್ಥರಾದ ಡಾಕ್ಟರ್ ಚಂದ್ರಾಯಪ್ಪರವರು ಹಾಗೂ ಡಾಕ್ಟರ್ ಸವಿತಾ ಮೇಡಮ್ ಅವರು ಉಪಸ್ಥಿತರಿದ್ದಾರೆ. ಅವರಿಗೂ ಕೂಡ ವಂದನೆಗಳು. ಸೊ ಈ ರೀತಿಯ ದೊಡ್ಡ ಕಾರ್ಯಕ್ರಮ ತಂಡದ ಶ್ರಮವಿಲ್ಲದೆ ಸಾಧ್ಯವಿಲ್ಲ. ಈ ಕಾರ್ಯಕ್ರಮದ ಸಂಯೋಜಕರಾದ ಡಾಕ್ಟರ್ ಲಕ್ಷ್ಮೀಕಾಂತ್ ಅವರ ಸೂಕ್ಷ್ಮ ಯೋಜನೆ ಮತ್ತು ನಿರಂತರ ಮಾರ್ಗದರ್ಶನಕ್ಕಾಗಿ ವಿಶೇಷ ಧನ್ಯವಾದಗಳು. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್ ಸರ್. ಎಲ್ಲ ವಿಭಾಗದ ಮುಖ್ಯಸ್ಥರು, ಅಧ್ಯಾಪಕರು, ಸಮಿತಿಯ ಸದಸ್ಯರು ಮತ್ತು ವಿದ್ಯಾರ್ಥಿ ಸ್ವಯಂ ಸೇವಕರಿಗೆ ಅವರ ಅಪ್ರತಿಮ ಶ್ರಮಕ್ಕಾಗಿ ನನ್ನ ಹೃತ್ಪೂರ್ವಕ ವಂದನೆಗಳು. ವಿವಿಧ ಪ್ರತಿಷ್ಠಿತ ಸಂಸ್ಥೆಗಳಿಂದ ಆಗಮಿಸಿದ ಅಕಾಡೆಮಿಷಿಯನ್ಗಳು, ಸಂಶೋಧಕರು, ವಿದ್ಯಾರ್ಥಿಗಳು ಹಾಗೂ ಭಾಗವಹಿಸಿದ ಎಲ್ಲ ಬೋಧಕೇತರ ಸಿಬ್ಬಂದಿ, ತಾಂತ್ರಿಕ ತಂಡ, ಮಾಧ್ಯಮ ಸಹಭಾಗಿಗಳು ಮತ್ತು ನಮ್ಮ ಪತ್ರಿಕೋದ್ಯಮ ವಿದ್ಯಾರ್ಥಿಗಳಾದ ಫರ್ದಿನಂಡ್ ಟೀಮ್ ಅವರಿಗೂ ಕೂಡ ನಮ್ಮ ಧನ್ಯವಾದಗಳು. ಸೊ ನಮ್ಮ ವಿದ್ಯಾರ್ಥಿ ಪ್ರೇಕ್ಷಕರಿಗೆ ವಿಶೇಷವಾದ ಧನ್ಯವಾದಗಳು. ನಿಮ್ಮ ಉತ್ಸಾಹ ಮತ್ತು ಬೆಂಬಲ ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಯಶಸ್ವಿಯಾಗಿ ಮಾಡಿದೆ. ನಿಮಗೆ ಹೇಳಿದ್ದು ಜೋರಾಗಿ ಚಪ್ಪಾಳೆ ಹೊಡೆದು. ಸೊ ಎಲ್ಲರಿಗೂ ಈ ಕಾರ್ಯಕ್ರಮದ ಯಶಸ್ವಿಯಾಗಿ ಕೈ ಜೋಡಿಸಿದ ಪ್ರತಿಯೊಬ್ಬರಿಗೂ ನನ್ನ ಹೃದಯಪೂರ್ವಕ ಧನ್ಯವಾದಗಳು. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್.
Thank you, sir. With this, we come to the end of today's valedictory ceremony. May I request everyone to kindly rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंधु गुजरात मराठा द्राविड़ उत्कल गंगा, विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा। तब शुभ नामे जागे, तब शुभ आशीष मांगे, गाहे तब जय गाथा। जन गण मंगल दायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
Thank you all for your presence and active participation. You can join us for the high tea. Also, the prize winners kindly come and meet me near the stage. Prize winners kindly come and meet me. And all of you stay back for the group photo in front of the KSTA main gate. Entrance, KST entrance.